Today, I want us to focus our attention on the ABO blood group system. There are a number of blood group system, but the ABO blood group system is the one that we are most concerned about in the general terms. However, there are other blood group system which are clinically significant, which we'll be getting into in a few moments. But when we're talking about the blood group system, I want you to bear in mind that we are trying to communicate the predominant blood that we see in the body, in circulation, the system of ensuring that oxygen is reaching the vital and critical organs and systems for the optimal functionality. So that is the system that we're talking about. We're not talking about the white blood cell. We're not talking about any other blood cell. We're talking about the red blood cell when we're talking about the blood group system in this segment. So the ABO blood group system is one of the most important of all the blood group system in both transfusion and transplant medicine. It is only blood group system that we use to identify and distinguish between individuals, and we can determine who has what antibody, who has what antigen, based on the presence or absence on the red blood cell. Without prior exposure, an individual has an ABO system, in that some antibodies are developed based on exposure to a antigenic uh, influenza. It may be bacterial, it may be a transfusion, it may be someone else's blood or something of that nature can generate different antigenic responses which change and modify your blood group observation. But the blood group system in and of itself remains the same or constant from birth till departure in this realm, right? So we have seen persons who have in pregnancy have some change in how their blood group system works. Different situations can cause undue change in the blood group system operation, which we'll get into in a little while. But the most important thing that I want you to know is that if you don't know any other blood group system, the blood group system that you need to know is the ABO blood group system. And then the question may be asked, what is your blood type? So when people ask what is your blood type, they're usually referring specifically to this blood group system. Are you A? Are you B? Are you O? Or are you even AB? But that system is different from the resource system, which is the D, which is where we said, are you A positive? Are you A negative? So the positive and the negative would be the resource. And the name rhesus was because it was derived from a rhesus monkey investigation. So that is why we have these different systems, and the systems are there to aid in ensuring that the blood that is transfused to a patient is beneficial. It is there to ensure that the organ that is transplanted to a patient will operate in the way that it was intended to operate, and we won't have graft versus host disease setting in, or we can reduce and eliminate most of those adverse outcomes. So we need to understand the blood group system for the ABO. And when you're thinking about the ABO blood group system, I want you to think about whatever is in the forward cannot be in the reverse. All right. So if the person is A, they that means that they have on the red blood cell there is the A antigen binding site, which means that they could not have A antibodies, because A antigen and A antibody comes together and form agglutination, and that would be devastating. So that there is a mismatch. So in that situation, you know that the antibody in circulation that we're looking for would be the opposite. That is the only way a person can live normally. All right. So if the person has A antigens, we are expecting them to have B antibodies. If a person has B antigens on the cells, the red blood cells, we're expecting in circulation them for them to have D. That's correct. So they're B, so they have A, they have the opposite. All right. So let us get a little bit more complicated. If a person has O, what is taking place? The person has O antigens, which means that they don't have A nor B on the red blood cell. That means they will have both A and B in circulation. All right. Simple as that. Now, if a person has AB, both A and B antigens on the red blood cell, what are we thinking? It means to us then, if it's all in the opposite, it's all in the reverse, we are thinking that they have no A antibodies and no B antibodies. And if you thought that, then you'd be perfectly correct. So this system is so simple that we can understand it, others can understand it, nurses can understand it, the physician can understand it and appreciate it. So this is what we normally use in the initial stages of determining someone's blood transfusion viability and the availability of bloods that are available. All right. And after we have done that, we would move now on to an IAT situation where we're trying to find out are there any active antibodies uh, taking over, which could complicate the reaction. And if the reaction is complicated, then we will not be able to just transfuse based on a room temperature crossmatch. We would have to do an immunoglobulin crossmatch. But before we can do the immunoglobulin crossmatch, we have to find out first what antibodies are causing the reaction that are not expected. So they are unexpected reactions, right? Because it's not following the normal protocol. And in those cases, the blood delivery may take a little longer. All right. So based on what we have discussed, you know a person who is O is considered to be the universal donor. All right. Which means that when we're talking about O, we're talking about the cells, which means that we have extracted the plasma portion or the liquid portion from the blood itself, which means that those circulating antibodies would be too low to create or evoke a significant reaction. All right. So the person who can receive from anybody based on what they have in circulation is a AB individual. So they are the universal recipient. All right, they have both A and B antibodies. So they can receive from a person who is A, they can receive from a person who is B, they can receive from a person who is O, they can also receive from a person who is AB. That is for the blood portion of it, the red blood cell section. When it comes on now to the plasma portion or the FFP or the cryoprecipitate, we have to be a little bit more specific. So let's talk about the FFP. When it comes down to the FFP, based on the fact that they have no A's and no B antibodies in circulation, we don't want to put anything in their circulation, because if we do that, then they will have a transfusion reaction. They would have an adverse reaction, which can cause the termination of their existence in this reality, and we don't want that. All right, so when we think about things from this perspective, we are now positioned to engage with the ABO blood group system. All right, so it's that simple, but there are some caveats that we need to understand in order to better prepare ourselves to dissect the other blood group systems. But for now, this is a brief introduction overview. So I hope that it was enlightening, it was insightful. And the question that you may be asking yourself is, what is my blood group? If you don't know your blood group, I think it would be a good idea to figure out what your blood group is. If you are a O negative person, it means that you are the universal donor. You can give to anybody your red blood cell. We're talking about the packed cells, not the whole blood. All right. In a situation where they need blood and blood products, especially if their hemoglobin is low, they're bleeding out, and we don't know the blood group, and your health history, which means that you don't have any disease that can be transmitted through a blood transfusion situation, then you'll be good to go. And if you think that you're a healthy person, it would be advised that you will donate blood, so that blood will always be available in circulation. And depending on which jurisdiction you are in, when you donate blood, you will know how many lives that one unit that you have donated would have saved. Greetings and welcome once again to another session. Today we're going to be continuing our conversation in relation to the ABO system. In the last discourse, we established that the ABO blood group system is the most important of all the blood group system, and we established that it was important in both transfusion and transplant medicine. All right, and we also disclosed that it is the only blood group system in which individuals already have antibodies in their serums to antigen that are absent from their red blood cell without prior exposure to red blood cell through transfusion or pregnancy. So this basically means that this blood group system, majority of the population is born with it. All right, and if you don't have it, then you are placed in another category, which is the look-alike to the ABO, which we classify as the Bombay. Due to the presence of these antibodies, transfusion incompatibilities can be derived. So if someone who is A gets B, that's a problem. If someone who is B gets A, that's still a problem. If someone who is O gets A, gets B, gets AB, that there is a problem. If someone who is uh, of a blood group that they are receiving from someone else who is not of the same blood group, that can cause some conflict, and there is going to be antigen antibody complex formation, because the body is going to try to protect itself, and in protecting itself, we're going to see a situation where the body's defense system goes on attack. And when it goes on attack, it creates these clump, which we call agglutination. And these things may result in blood clots form. But what happened at the end of it all is that it causes the organism to come to its demise. So we have to be very careful and mindful in ensuring that the blood that is transfused is one that is viable, one that is suitable for the purpose intended. So, ABO blood group system is a system that you want to be most proficient in, because this ABO testing is done in almost all blood banks. All right, this is the first step in determining a person's uh, viability for transfusion. All right, so even today, transfusion can go wrong if there is an ABO incompatibility. All right, so we have to just bear this in mind. So we have Carl Lanster to thank for the ABO blood group system, at least based on documented evidence. It was in 1901 that Langster drew blood from himself and 5 associates. He separated the cells and the serum and then mixed each cell sample with each serum. So when we're thinking about him mixing the cells and the serum, we're thinking about the forward and the reverse blood grouping. The forward blood grouping is the front grouping, is defined as using a known source of commercial anti-serum, which is the anti-A and the anti-B, all right, to detect antigens on the individual's red blood cell. Because remember, the red blood cells, let's bring it up. The red blood cell, we're going to put it there. This is the red blood cell that we're looking at. On the surface of the red blood cell, these are the antigen binding site that we have right here. These are the antigen binding site, and each of them have their own specific look. All right, and that is why we have the goodness to fit, because specific antigens bind with specific antibodies. All right, very good. So now the reverse blood grouping, or the back typing, is what we considered to be the method of detecting ABO antibodies in the patient's serum by using reagent red blood cell. All right, so we are doing the opposite. And basically, we normally use the A1 cells and the B cell, but there are different subgroups of A's, and depend on where your testing is done, depend on what the predominant blood grouping is, then you may have to use the subgroups like the A2 or the A3. All right, so it all varies, but the idea is that you're going to have A cells and B cells in order to determine what anti antibodies are present. All right, so the A cells and the B cells give us an idea so that we can compare it with the forward grouping, because remember what we established first. If they have A antigens, then they will not have A antibodies, because if they have A and A, it's going to come together and it's going to form agglutination, and then that will lead to the termination of that individual's existence. So if the person has A, we expect them to have A antigens, we expect them to have B antibodies in circulation. If the person has B antigens, we expect them to have A antibodies in circulation. If the person has no antigens on the cell or their own, we expect them to have both A and B in circulation. If the person have both A and B antigens on the surface of the cell, then we expect them in circulation to have no antibodies. All right, so that is just an idea as to how things work. So let us go back to it. This is a cell, and on the cells we have the antigen binding site, and each antigen has its own unique look. So the A people have a specific look for the A, the B have the specific look for the B, and that is just how it is, right? So we have established the forward blood grouping, and we have established the reverse blood grouping. So these two blood grouping are still a part of the ABO blood grouping system, and that is just one blood grouping system. There are other blood grouping systems that are implicated in transfusion reaction, but they are not necessarily at the room temperature in which they are identified. They have to be taken through some other testing methodologies to determine compatibility. And we are going to go into those in a little while, but we just want to focus on the ABO blood group system. So ABO blood group is the most frequently performed testing in the blood bank. It's the most frequently performed testing. Both the ABO forward and reverse grouping testing must be performed on all donors and patient. So the donor has to be tested, the patient has to be tested, because this is the simplest form of compatibility. We want A people to receive A blood, B people to receive B blood, O people to receive O blood, AB people to receive AB blood as best as possible. But there are instances where we can give them different blood based on what is available and what is compatible. All right, because you know that, let us go back on our board. An O person can receive blood from an O individual, right? A B person can receive blood from a B person or an O person. That is the packed cell that we're talking about, because remember, the O person has no A or B antigens on their cells, so they can give the red cells to a person who is B. A person who is A can also receive blood from a person who is A and O. That is correct. So we're doing very good. Now, this is where the caveat comes in. A person who is A and B can receive from a person who is A, can receive from a person who is B, and can receive from a person who is O. And you're saying, wow, this there then puts this individual as the universal recipient. So the AB person can receive blood from any of the blood groups. The O person, we realize that O is right here, O is right here, O is right here, O is right here. So this O person can be considered to be the universal donor. So the O person is the universal donor, which means that they can give to anybody. And the AB person is the universal recipient, which means they can receive blood, the packed cell portion of the blood, from anyone. All right? That's all we need to bear in mind in this moment in time. Now, as we move further along into the ABO blood grouping system, it has been postulated that in some instances, bacteria, pollen particles, and other substances present in nature uh, are sometimes chemically similar to the A and the B antigens, right? So bacteria are widespread in the environment, which constantly uh, can create, in some instances, some exposure and present some A like observation or B like
antigens being observed, this exposure serves as a source of what we call ABO discrepancy sometimes. Because a person who is A should have a particular blood group in the reverse observation. A person who is B supposed to have a particular in the reverse observation. But sometimes environmental factors can cause someone who is O to look like someone who is A. So then there is some incompatibilities with the forward and the reverse, which would lead to further investigation to determine what is the cause of this situation. All right, so we just have to bear that in mind. All other defined blood group system do not regularly have naturally occurring situation taking place. They are usually based on exposure to something, right? And the body then produce the antibodies based on the reaction or the interaction with the antigens. All right, so just bear that in mind as we move further. Let us talk about the ABO antibodies. Individuals normally produce antibodies directed against the A and or the B antigen present on their red blood cells. So basically, as we established before, if you are A, you're going to have the opposite, which would be B antibodies in circulation. If you have the A antigens, you have the B antibodies. If you have the B antigens on the cells, you're going to have the A antibodies in circulation. So this is just the natural way how we have been designed to preserve our existence. So let us just bear that in mind. So let us move forward now and understand that the ABO antibodies are predominantly IgM. Yes, and they are activated by complement and they react at room temperature and can even react in cold environments, right? So ABO antibodies produce strong direct agglutination reaction during the ABO testing. ABO antibodies produced is initiated at birth. However, though we are born with all of this, when we are under 6 months, the body has not yet mounted uh, enough titer or enough concentration to give us a reliable and reproducible uh, result when this testing is performed. So what we do for the babies and for the geriatric is sometimes for the babies we do not perform the reverse blood grouping if the individual is under 6 months. For the adults, geriatrics particularly, when they are over a certain age, like they are getting to 70, 80, 90, 100, when they're in that age, we tend to find that there is a decrease in the reactivity in the reverse blood grouping. And this there is usually due to the fact that as we get older, our antibody production is reduced. And because of that reduction, we may have some challenge in confirming the forward blood grouping with the reverse blood grouping. But understanding the patient history gives us an idea as to what are possible causes of the discrepancy that is being observed. So to perform the ABO blood grouping system, we need the anti-A reagents, we need the anti-B reagents, we need the A cell, usually the A1 cell, and we need the B cell. All right. So the A reagent is usually monoclonal, it's highly specific, it's usually IgM, and the color that is used to denote the A is a blue coloration. The expected reaction is usually 3 to 4, and we usually do 1 to 2 drop depending on your laboratory directions and the operation procedures which they are going by. All right. So for the anti-B, these are usually monoclonal antibodies. Yes, they are highly specific. They're IgM as well. The color though for the B reagent is usually yellow, and it contains a dye. The dye is called archiflavin dye. So that is the dye that it contains. All right. So the expected reaction still is 3 plus or 4 plus, and usually it's the same 1 to 2 drops that are used. For the reverse blood group in the A1 and the B cells, it is usually derived from human sources. So persons who are A, they are the ones who we get the A cells from. But we usually use the A1 lectin to determine persons who are A1, and we get the blood from that source and pool them together. But in some laboratories, there are commercially prepared A1 cells, which makes it easier for the laboratory to operate. So they don't have to be preparing their cells to use their cells. They can just source their cells and have them quality controlled from the preparer's laboratory while they just use and enjoy the benefits of the cells. All right. So we usually do 3 to 5% suspension. Some may say 4 to 5% suspension, but it varies based on the location, based on the concentration, based on the preparation. So the expected reaction with the A1 cells and the B cells is usually from 2 plus to 4 plus, and it's usually 1 drop that is utilized. All right. So let us just do a quick summary of the forward patient cells with the reagents. So let us go back on our board once more. So let's say the blood group, and we're going to say anti-sera. This is for anti-A. Then we're going to say this is anti-sera B, and we're going to come to the conclusion of the antigens on RBC, red blood cells. All right. So a person who is O, we are expecting no reaction with the A, no reaction with the B, right? Because there are no A or B on the red blood cells. So there is none, all right? A person who is now A, we're expecting a reaction in A, no reaction in B, because this person has A antigen. Now, a person who is B, so we get in the picture. We're expecting no reaction with the A, we're expecting a reaction with the B, and it is because there is B antigens. Now, a person who is AB, we're expecting a mixture of reactions. We're expecting a reaction with the A, and we have a reaction with the B as expected, because this person has both the A and the B available. All right. So with that observation, with that understanding, we have an appreciation as to what is taking place. Now, this is a forward. Let us write it forward. FORWARD. So this is a forward blood grouping. Now, let's move to the reverse blood grouping. All right, so this is the reverse, REVERSE, blood group. All right, so we're going to be using cells in this instance. So we have the A1 cells and we have the B cells. And this is going to give us which antibody or bodies exists in the serum, right? So the person is O. We're expecting to have a 4+ reaction in the A cells, a 4+ reaction in the B cell, because there are A and B antibodies in circulation. A person who is A, we're expecting no reaction with the A, but we're expecting some form of reaction with the B because there is B in circulation. When a person is B, we're expecting a reaction in the A, but no reaction in the B because they have A. Now, when a person is AB, we're expecting no reaction in the A and no reaction in the B. So we are going to put none. All right, so when we're looking at this now, we're seeing none, none. So this here, for the AB person, this, whatever liquid portion of the cell that exists, it can be given to everybody. All right, so that is just something that we need to understand. When a person is AB, they can give their plasma portion to anyone. And that is just something that we have to understand and appreciate. All right, so that is in a nutshell how the ABO blood group system is. But I must bring to your attention that when it comes on to the phenotyping frequency, there is a different ethnicity that has predominant in certain blood groups. All right, so let us talk about the phenotype. The phenotype just basically denotes the O, the A, the B, and the AB, right? That's the phenotype, what we observe, right? So a white person or a Caucasian person, then we have a black person, we have the Hispanic, and we have the Asians. We're observing that 45% approximately of the O exists in white population. The white population, 40% A, then 11% B, 4% AB, right? For the blacks, we have 50% O, so majority of the black population is O. Then we have 26% which being A, then we have 20% being B, and we have 4% being AB, just as in the white population. So the black and whites have the same AB ratio. Now for the Hispanics, we have more persons, 56% being O, and then we have 31% being A, we have 10% being B, and we have 3% being AB, right? Now for the Asians, we have 40% being O, we have 28% being A, 25% approximately being B, and AB is 7%. So we are realizing that the Asian population have more AB, while the one with the lowest would be, for the AB, would be the Hispanic population. In the same breath, we realize that majority of Hispanic individuals will be O, all right? But when it comes on to O blood group, we are only expecting 40% of the Asian population. So this is just something that we need to know and just have an idea. So when you go to the physician or when you go to the provider and they indicate to you that they need to know your race and ethnicity, it's not always because there is some discrimination taking place. Sometimes when it comes on to a medical procedure, it just gives an initial overview as to what can be expected. And when this information is derived, your treatment can be tailored in such a way that it is beneficial to you and it provides you the best opportunity for a fast and speedy recovery. All right? So just bear those information in mind. We could go into the offspring determination for the ABO, but we're going to leave that for just a little moment and understand that when a person has A blood group, they have a particular marker, right? When a person has B, they also has a particular marker. And when we say marker, we're talking about sugar because these are just sugars, right? When a person is O, we classify them as H. They also have a particular marker, all right? So a person who is A has this marker. A person who is B has this marker. And a person who is O or H has this marker. And as it relates to the immunologic sugar or the immunodominant sugar, a person who is H, the sugar is L-fructose. The person who is A, the sugar is N-acetyl D-galactosamine. All right? And then a person who is B, it's D-galactose, all right? Galactose. So this is just something that we need to understand, and it's because they have the different antigen. So based on which antigen they have, and then this here is the immunodominant sugar. So these are just some things that we need to understand as it relates to the ABO blood group system. So we need to go into the sugar a little bit more so that we can have a better understanding and appreciation. But we're going to take this up in our next sitting because, as you know, class is dismissed. Until we meet again, continue to study, continue to show yourself, approve. And at the end of the day, when the results have been determined, your results will be indicative of the invested time that you would have deployed into learning this material. We're going to go through some cases and some questions that are used to gear you for the examination, uh, the ASCP examination that is a possibility for majority of those of you who are observing and understanding. It is my intention to provide a resource, but before I provide the questions, I want to provide you some guidance in theory so that you can use the information you receive to answer the said question and apply the knowledge as is necessary.
Let us talk about how people inherit the ABO blood group system. So, the theory for the inheritance of the ABO blood group system was described first in 1924 by indicating that an individual inherits one ABO gene from each parent because two parents normally come together to produce a child. So 50-50. All right. The inheritance of the ABO gene therefore follows the Mendelian genetics. So ABO, like most other blood group system, is codominantly expressed. So codominantly expressed means that it is co in partnership with, all right, one position or locus on each chromosome 9 is occupied by A, B, or O gene. The O gene is considered an amorphous as no detectable antigen is produced in response to the inheritance of this gene. So remember, when a person is O, this person does not have any A or B antigens on their red blood cells. So this is what gives it its amorphous uh, definition or distinction. So the group O phenotype is an autosomal recessive trait with the inheritance of two non-functioning O gene. And in order to be O, you have to have OO and OO. All right? So both genes non-functioning, so we call it the auto-recessive. The designation group for the A and B refer to A and B phenotype. You can be AA, you can be, yeah, for A, you can be AB, and for B, you can be BB, you can be BO, and you can be AB. All right? And for the A, we missed one. You can also be AO. So The designation group of A and B refers to the phenotype where AA, BO, and OO denotes the genotype. In the case an individual is O, both phenotype and genotype are basically the same because the individual would have inherited a homozygous for the O gene, because in order to express O, the person has to have OO from one parent and OO from the other parent. An individual who has a phenotype of A or B can have a genotype of AA or AO. And for a B person, that's for the A, and for the B person, you can have BB or BO. So we're going to just try to wrap our minds around the genotype and the phenotype so we can have a basic understanding of how the blood group system is derived. So let us get this on our screen. So we're going to start off with having the offspring possible genotype. Offspring possible genotype. Well, let's go with the phenotype first. And then we're going to do the mating genotype. And then after that, we're going to have the mating phenotype and genotype. All right, so possible outcomes. So the person is A. We can expect one parent to be AA. We could also expect another parent to be AA. All right, a person is one parent is AA, another parent can be AO, because AO also gives you an A. One parent could be AO and another parent could be AO. All right, so that is basically what is happening. So this person is A with AA. This person is A with AA for the first set, or AO. This person is also A with AA or AO, or these two O's can come together and then give us the person group to be O, which indicates to us if they are O, O can only be expressed by OO. All right, so that is basically what happens. If a person is B, multiply by B, we're expecting the same BB times BB, and then we have BB times BO, and then we have BO times BO, and then the breakdown is the same. So this person would be B, so you'll have BB. This person is still B, you'll have BB. That's a possibility, or the person can be BO. And this is basically the combination BB BO. That's basically it. This person is B, so you have B, B. This person can be B, O, because we have BO, BO, all right? And this person has O and O, so there is a possibility that this person could be an O, all right? So that there exists for this individual. And if we do the other groups, we can realize how they play out if we are mating with different persons. So if an AB person, let's try to put that on the screen. Let's see if we can get some space. All right, so we're going with a AB person. AB, I hope that this is bright enough. Let me see if we can get a little bit bigger. Let's go back. Let's erase this. And let's try again. So a AB individual with a AB individual would give us one thing. We can get A, B, and A, B, because A and B, B and A, it's AB. It's the same thing, all right? So this person has a lot of potential. This person can be A, 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 A. This person can be AB, AB, all right? So this is one, this is two. This person can also be BB. However, we should note that this person will never be O, because there is no O recessive to give us the O. So none of the parents have O recessive, so we can't have an O individual. But if even one parent had an O recessive in this grouping, we would also need another parent to have a recessive O in order for us to have the expressed O. All right? So that is just something. Let us try the O and see O person times O person. In order to be an O person, the only thing that you can have is O, O, and O, O. And if you have OO and OO, then all you can have is O. All right? So that is basically what is happening. All right? And this here would be for the A. This is A. This is A. This is also A. Or AB. This is A. This is AB. And this is B. All right? So we have that clear up. So let us look at mating with someone who is of a different blood group system. So if your partner or you may be A, let's go. So you're A and your partner is probably B. What do we expect? So this one person is going to have AA. That is a possibility, right? The other person may be BB. That's a possibility. Then if that person is A, this person can have AO, and this person can have BB. This person, one person can have AO, and the other person can also have BO. So we have a variety taking place right here, all right? So we have one person being AA times BB, or AO times BB, or AO times BO.
But we also could have a situation where we have one person being AA times BO. All right? But all of these would be the combination of this, the mother and the father coming together. So this person has a potential to be a B individual. When an A person, a B person come together, the child has a potential to be an AB individual. When an A person and a B person come together, the child still has a potential to be an AB person, but there is also the probability or the possibility that this child can be, based on BO, can be a B person. And when that happens, we have an idea as to what the genotype is. All right, so BO. So we have an idea that one person is AO and the other person is BB. All right, then based on this combination, there is a possibility that one, a child could be AB. Based on the A and the B combination, the child could also be, guess what? This combination gives us something very unique. So AO times BO gives us a very unique combination. We're going to write this down here. AA times BO. We're going to carry this down here. That's what we just did. So we can have some space. I'm going to show you something right here. All right. So it's just expanded it. All right. So with this combination, we have AB, which is AB. We have OO, because if we combine this, we have O, because one is O and the other is O. So a AO and a BO can give a O individual because we have two O's that exist right there. All right. Then there's another combination that is possible from this. And this combination is, guess what? AO, because A and O. So you have A, which is AO. So if you have AO, there is a possibility that you can have BO, right? Because you have B and O. So for combination B, and you have this person is a BO individual. So now we have a better understanding and better appreciation of what does that mean. So this one we can review and go and deduce what is a possibility of the phenotype genotype relationship. So the genotype is AA times BO. So we are expecting to have at least AB, because A times B, all right? Then we are expecting to have A times O, so we can have AO, which is A. So this is AB and this is A. And then we can have, is there any other combination? There's a B that is present here, BA, which is AB. All right. So we have these two that are possible from an AA times a BO. So we have AB and AO. So this is the excitement that comes on when we're doing genetics. It's pretty straightforward if you understand the basic tenets of multiplication. Uh, that's basically what you have to do, and you have to understand that there are basically it's a ABO blood group system. So with that said, we have A, we have B, and we have O. All right, beautiful. So let us see if we can mix it up a little bit. So a person is, this is just for our edification, A and O. If a person is A and O in the phenotype, we are expecting the possible mating match could have been AA times OO. Or for the A, what else could give us A? We have AO. But the person is O, so the person has to be OO. Once a person is O, there is only OO that produces an O. All right. So from this, we can deduce that the person is going to be A, and this A that is going to be produced would be an AO in the genotype. So this person could also be an A, and from this A, what is going to be produced is an AO. But A times O give you AO, but you have O and O. So you have, this person has also the possibility of being an O. So you have OO. So this person can be an A or an O. Next, let us try a person who is A and they mated with an AB person. So in order to be A, we know you can be AA. In order to be A, you can be AO. All right, so those two give us A. Now, in order to be AB, all you can have is A and B and A and B. So based on this, we're going to deduce what are the possibilities. So this person could be an A based on A times A, which would be a solid A, AA. All right? A times A gives us a solid A. All right? But this person also could be A times B, which in this situation would be AB. And if we try to do it another way, these are the combinations we get. Now, this person can also be A, and this can be A times A, that type of A. This person can also be so you have AO. So we were going to look at it. This person is AO times AB. So this person can be A, and this A is AA or AO. This person can also be B, because the AB, which would be a BO, and this person can also be AB as expected. All right, so we have AA, we have AO, we also have BO, and we have our favorite AB. All right, so this is just an idea for us to understand how the offsprings come about with a different genotype based on the phenotypic expression. All right, so let us do a little bit more just for the euphoria that comes with it. Now that we have a better understanding and appreciation. So the person, two parents come together, one is B, one is O. In order to be B, you can be BB or you can be BO. In order to be O, you can be OO or OO. So based on this combination, we are expecting the person to be B, and this person is going to be BO. So B times O, B times O. So that's BO, right? This is a B, BO, all right? This person is BO or possibility BO or OO. So we have two things that's happening here. This person can be BO, bam, that's number one, or this person can be a O. And the reason B times O, O times O. So when there are two O's, that's the only time an individual can become a O person. All right. So that is beautiful. Now let us go to another. Let us try B. We have a B person and we're going to mesh them with an AB person. All right, so to be B, you can be B, B. To be B, you can also be B, O. And when you are AB, you can only be AB and AB. All right, so now B times A, we have AB. Just like that, right? And then if we come down a little further, we can see where we have B times B. So guess what? We have a B right here, a solid B taking place. All right, let me just give some room to this. Let's bring this down a little bit. So BO and AB. And then let us erase this so we can have some clearance. All right. So now that the person is BO or AB, what are some of the possibilities that we could expect? So a person who is BO, what we can expect is, in order to be B times A. So the first thing that we're looking at is AB, and it's because A times B. Then we have O times B. So 2, we are seeing B, we're having BO. All right. Are we getting that? But is there something else that we miss? We have B times B, which can also give us B, right? So B times B can also give us B. And is there anything else that we're missing? We have 2 possible combinations grouping. Is there any other possible? Remember, the person is AB, so we are going to see if there's an A that is possible. All right, is there an A that is possible? A times O would give you AO. So from a B person times an AB person with the genotype of BO and AB, we can have an AB person and we can have a B person with a BB or a BO, or we can have an A person with a AO. All right, beautiful. Now let us try a last one so that we can have a clear understanding as to what is really happening. All right, so this one is for you to do, to understand, to appreciate. All right, so a person is A, B, and one parent is O. What are the possible genotypes that we're looking at? With an AB person, in order to be AB, you can only have AB, right? And in order to be O, you can only have O. So now there are only 2 possible combination. You can have A, and the only way you can have A is if you have A times O, you have AO. You can have B, which is B times O, which would be BO. You cannot have any other combination because in order to express O, you have to have O recessive from one parent and O recessive from the other parent to come together to give a fully recessive expression. All right, so with a AB patient and a O patient, the only two combination is to split that AB to A and to O. So that is just an idea as to how we can understand and appreciate the blood grouping system and how the combination exists when it comes on to mating. This is the ABO group of offspring for the various possibilities of mating. So we have gone through all of them. So now you have a better understanding as to what your blood group could indicate about your parents. What are the possible combination? Now, there is something that we should be aware of. We have some types, and genotypes give rise to the expressed phenotype. And when we talk about the genotype, we're talking about the gene, and the phenotype is what is basically expressed, what is displayed. So your parents can be, if they're A1 and A1. All right. So the genotype that we're going to expect is A1. So the phenotype is A1, right? A parent can be A1, one can be A2. And what can we expect? We can expect A1, all right? So A1 and let's say we have O, we're going to still expect A1. If the genotype is A2 and A2, we're going to expect A2. If the genotype is A2 and O, we're going to expect A2. So A1 is stronger than A2, A2 is stronger than the O, all right? Just bear that in mind. So if we have A1 and a B, we can expect a A1B individual. If we have A2 and a B, we can expect a A2B individual, all right? And if we have a genotype in which there is OO, we're expecting, it's simple. For the phenotype, if we have BB, we're going to expect B. If we have BO, we're going to expect B. And we went through the rigors already. So you have a basic understanding as to how we move from the genotype into the phenotype. So that is just something that we want to cover in this sitting. In our next discourse, we will go into understanding the complexities associated with the formation of the A, B, and H red blood cell antigens. Let us talk about it. Let us talk about the ABO system. And while speaking about it, we are going to be speaking about the formation of the A, B, and H red blood cell antigen. Because on the surface of the cells, we have antigens. In circulation, we have antibodies. So we're going to consider it to be the formation of the ABH antigen, which is a result of an interaction between three separate loci: the ABO, the HH, capital H, lowercase h, and SE, capital S, and lowercase e. So these genes don't actually code for the production of the antigen, but what it does is, guess, it produces a special or specific glycosyl transferase that adds sugar to the base
precursor substance. All right, so in this interaction, we can have an appreciation that sugars are involved in the formation of the ABH on the red blood cell. So a paraglobulicide or a glycan is the same basic precursor material from which the A, B, and H antigens all originate. So all these antigens originate from these glycans. So specific enzyme transferase elicited by an inherited genes attaches to a paraglobin side or a glycan. And when this happens, we have the different resulting ABH antigen being expressed on the cell. So when terminal galactose on the precursor substance is attached to N-acetyl glucosamine in beta 1, there's a link to there's a 4-link attachment that is taking place, right? The precursor substance on erythrocytes is referred to as the type 2. So beta 1 move into giving us a 4 linkage, and then that leads us to type 2. So let us see if we can put this on our board. So we have beta, BETA, beta 1 goes to give us a 4 linkage. And then this now gives us type 2. All right, so the ABH antigen on the red blood cell are constructed on oligosaccharide chains of the type 2 precursor substance. So if we have type 2, I hope that you are thinking there must be a type 1. So type 1 would come before type 2. So let us get into type 1. A type 1 precursor is referred to as the beta 1. And this has 3 linkage between the galactose and the N-acetyl glucosamine. All right? And this generally gives us another type of formation. So let us just put what we have stated. So we have type 1 and we have type 2. So type 1 is what we're talking about right at this very moment. So for type 1 we're gonna have beta 1, which is going to link to 3. So we have 3 linkages. So 3, beta 1, and type 1. So for type 2, as we discussed before, we have beta 1, same beta 1, but this has 4 linkages. So I want you to observe. So 2, 1, 4 rather, 1 and 2. All right. So this is basically
type 1, and type 2. So the H antigen is a precursor structure on which A and B antigens are made. So in order for A or B to be made, we need the H antigen. And that is why the ABH system of antigen is important for us to have an appreciation of their formation. So, inherited based on our inheritance of the H gene, this will result in the formation of the H antigen. So you could have FUT1H or FUT2SE gene. All right. So these two genes are closely linked and located on chromosome 19. So let us put that down so we can have a visual as to what does that mean. So we have FUT1H and FUT2SE. All right, and these are on the chromosome 19. So just remember that, all right? In contrast to the ABO genes, so when we have the ABO genes, we discussed previously that this was located on chromosome 9. All right. So the H and SE are located on chromosome 19, while the ABO genes are located on chromosome 9. So the H and SE genes are not a part of the ABO system. All right, so we need to get that clear. Yet, there is the inheritance influence to the A and the B expression. So, though they are not directly associated, they do indirectly influence the formation of the ABO antigens on the red blood cell. Then this brings us to another idea that we need to be mindful of. All right, and we think about the SE genes. When we talk about the SE gene, which must be inherited to form the ABO in the secretions, I want you to get the two things. SE, secretions. When we think about secretions, we are thinking about secretors. So just bear that in mind as we traverse the exploration of the ABH formation. So the ABH antigen develops early in fetal life, but it does not increase much in strength during the gestational period. So the RBC of the newborn has an estimated carrying capacity of about 25 to 50% of the number of antigen sites found on an adult red blood cell. So this is basically giving us the idea that when we're doing a reaction, there is a possibility that an infant will have a lower reaction when we're grading it, like 1 plus agglutination, 2 plus, 3 plus, 4 plus. In the adults, you may expect a 4 plus or a 3 plus. For the infant, you may expect a 3 plus, a 2 plus, or even a 1 plus based on the amount that is being expressed. All right, so just bear that concept in mind, so that this will give you a guide or give you insight as to how different markers are expressed, and we can take advantage of this when we're doing our ABO discrepancy assessment. So this is important for us to know. And now that you know, let us move along. So consequently, reaction of the newborn red blood cell with ABO reagent antiserum are frequently weaker. All right. The expression of A and B antigen on the red blood cell only fully develop at about 2 to 4 years of age. And when it is fully developed at that time, it remains constant throughout the rest of the life. However, in some instances, when a person becomes a geriatric, the body starts to produce less antibodies. The forward will remain uh, constant, the reverse, which developed later based on the antibody expression, because for the babies, as we discussed before, we don't perform the reverse blood grouping. So too, when we go back into a geriatric state, what happens is that the body's ability to produce antibodies is reduced, and this reduction gives us lower grading or difficulty in identifying the antibodies because the titer is so low. All right. So you were in as a child, you will be when you become a geriatric. So it's the same kind of systematic processes that take place. So in addition to this, we need to understand that the phenotype expression of the ABH may vary with race, genetic interactions, and disease state. I know that when we speak about race, many persons are apprehensive to disclose their identity, but when it comes on to the medical field, understanding where a person is derived from, the race of the person, their genetic markers, their disease state, helps us as practitioners to come to a realization as to what are the possible causes of the condition which impales the individual. So when you're asked about your race, don't feel apprehensive or hesitant to disclose it appropriately. All right. Now let us look at the glycosyltransferase and the dominant or the immunodominant sugars responsible for the H, A, and B antigen specificity. All right. So let us put the gene, Gene, all right. Then we're going to look at the glyco, Glycosyltransferase, all right. Then we're going to have, maybe I shouldn't write this out. Let me make some more glycosyl. We're going to put transferase down the bottom, but this is one word, transferase. All right, but that is one word. Then we're going to talk about the immunodominant sugar, immunodominant sugar, and then we're going to talk about the antigen, ANTIGEN. All right, I hope that we can make this out. So we're going to talk about the antigen. Let's write that. A-N-T-I-G-I-N. T-I-G-E-N. All right. Let us draw a line, draw a line, draw a line. All right. So the first we know is the ABH system that we're talking about. So let us talk about the H. When we talk about H, we are thinking the gene FUT1, right? So in order to get FUT1, the glycosyl transferase that we need is two. Well, let's start with the alpha. It's alpha 2 L, and it is fructosyl transferase. FUCOSIL transferase. All right. And then this here, the immunodominant sugar would be the fructose. All right. So don't forget the L that we have. So it should be L fructose. FUCO fructose. All right. And then this here would give us the gene. So the antigen that is going to be expressed is H. Simple. So that is the hardest one. Now for the easier ones, A. If a person is A, they're going to have alpha, and this is going to be 3. And not only 3, 3N. And then now we have acetyl galactosamine transferase, or some may say acetyl galactosaminal transferase. All right. So anyhow you want to pronounce it, as is applicable in your jurisdiction. But what I'm going to do is spell it out so that you can have it documented. So it is alpha 3N ACYL All right. So that is it. So when we're moving to the immunodominant sugar, we have an N here. So we're going to put by the N, and the sugar is acetyl D galactosamine. All right. So it's N, and we use the same name. And remember, we started with A, so we are going to end with A. Beautiful. Now, let's go to B. I don't want you to be intimidated by the glycosyl transferase. I just want you to be able to identify them and understand the sequence of operation. All right, so you can know the immunodominant sugar, which are easier to pronounce, enunciate, and you can know the antigen because those are simple. So it's based on the ABC free sequence, but instead of ABO, we have ABH. All right. So now we move into B. So B is going to be alpha, and just like the A, it's going to be 3. However, when it comes on to the letter, it's not going to be L or N, it's going to be D. And for B, it's going to be galactosyl transferase. All right, so GALACTOSYLTRANS ferrase. All right, so we're going to be putting the immunodominant sugar, and remember the D, we put back that D, and it's galactosyl. Where does it come from? Galactose. All right, so we're going to write GALACTOSE, and then this, now we started with B, so the antigen that we expect to be expressed is B. Beautiful. So that there is exactly how the operation and the association with the glycosyl transferase and immunodominant sugar are expressed. So this is just something that we need to be aware of so that we can identify, and then we can explain as is needed. All right. So what I want you to know is that there's an interaction with the HH, capital H, lowercase h, and the ABO genes. So individuals who are blood group O inherit at least one FU1H genes, or its genotype HH, capital H, or H, capital H, lowercase h, and 2 O genes, because you know that to express O, you have to be O. All right, so if you are AO, you're not going to express O, you're going to express A, but you can transfer the O and you can also transfer the A to the child that is going to be derived. All right, so just bear those concepts in mind. So the H genes elicit the production of an enzyme called alpha 2L fructosyl transferase that is that transfers the sugar L-fructose to an oligosaccharide chain on the terminal galactose of the type 2 chain. So basically, this is explaining to us the interaction that takes place and how the ABO system interacts with the H system, so as we can have a mental visual picture. Therefore, let us consider the L-fructose is a sugar that is responsible for H specificity. The O genes at the ABO loci is sometimes referred to as an amorphous, and does not elicit the production of a catalytically active polypeptide transferase. Therefore, the H substance remain unmodified. All right, so this is what we need to understand for the O portion of the blood group system. There is a O lookalike that is referred to as the Bombay. We're going to have to discuss that a little bit further so that you can be able to differentiate genotypically an O individual in comparison to a Bombay individual. All right. The H genes is present on 99.99% of random population. Its allele lowercase h is quite rare. And the genotype HH, which is lowercase h, is extremely rare. And this is where we get the Bombay term to be applicable. So the term Bombay has been used to refer to the phenotype that lacks the normal expression of the ABH antigen because of the heredity or the inheritance of the lowercase h genotype. So the lowercase h genotype does not elicit the production of alpha 2 L fructosyl transferase. All right. So accordingly, the L fructose is not added to the type 2 chain, and the A substance is not expressed on the red blood cells. So we have no H being expressed. And you know that we need the H to give us the A or the B. So when that is not being expressed, then we have a problem. It is a problem because majority of the population doesn't have the Bombay type. Because majority of the population, 99.99%, has a capital H somewhere. So they have capital H, or capital H, lowercase h. The lowercase h is the Bombay type, which is the 0.1% of the population. All right. So when we are thinking about this, it's a problem because not that we can't identify, the problem exists because when we are seeking after blood for these individuals, it is arduous, especially if we don't have a population pool of individuals who have this blood group. So even though the Bombay HH individual may inherit the ABO gene, the normal expression as a result in the formation of the ABH antigen does not occur. It does not occur. So this individual, the Bombay, is phenotype OH. So that's O and subscript H. Let me just write it just in case you come across it. So it's O and subscript H. This here is the BAM. No, it's actually Bombay. So it's BOMBAY. All right. And the expression is HH. So this is not blood group O. I want you to remember this. If you see this, this is not blood group O. It looks like O. It's observed in the forward as O, but this is not O. All right. Now, in the formation of blood group A, what happens is that the A gene, and you know that we stated that if a person is A, they have the AG. They can be AA or AO. So this situation codes for the alpha 3 N acetyl galactosamine transferase, which transfers the N acetyl D-galactosamine sugar to the H substance. So when this transfer is done, the sugar confers A specificity. Simple as that. So all the information that we had previously is now coming back full circle. So we are seeing how it plays out. So the A gene tends to elicit a higher concentration of transferase than the B genes, leading to conversion of nearly all the H antigen on the red blood cell to A antigen sites. As many as 810,000 compared to 1,170,000 antigen sites exists on an A1 adult red blood cell in response to inherited gene. So we're going through this. I want you to also be thinking in your mind, when a person is A, we realize that they have more expression of A than when a person is B. So let us picture in our mind a person is AB. If we don't have AB available, the first alternative, if we're going to choose between A or B to give to the person, is going to be the one that is mostly expressed, which would be the A. And because A is usually predominant in most population, A would be more available for the transfusion process. So these are things that I want you to put together in your mind so as you can have an appreciation for the whole flow of things. Now let us get to individuals with the blood group B. So this is inherited. An individual with B can be BB, capital B, or it can be BO. All right, so these genes code for alpha 3 D galactosyl transferase. All right, and this attaches the D galactose sugar to the H. And when this does, there is a situation that is placed or considered the type 2 precursor substance through which the action of H can generate the B. And that there is how we get, in essence, the B being expressed. This sugar is responsible for the B specificity. Anywhere from 610,000 to 830,000 B antigen site exists on a B adult in response to the conversion of the H antigen by the alpha 3 D galactosyl transferase to produced by the B gene. So if you have the B gene, then you know which sugar was implicated in the expression. So let us go back now. When both A and B genes are inherited, the B enzyme seems to completely try its best to compete more efficiently for the H substances than the A enzyme. As a result of this enzyme competition, the average number of A antigen on the AB cell is approximately 600,000 sites compared to an average of 720,000 B antigen site. So the interaction of HH and ABO gene uh, can be observed. So this is a good place to pause as we allow a moment in time to digest the information that has just been presented to us. As we have covered in brief the interaction of the H, capital H, lowercase h, and the ABO gene. So we have established the gene, we have established what is the glycosyl transferase, we have established the immunodominant sugar, and we have established what is expressed. We have also identified some of the potential sites expression in what quantity, so that we can have a mental picture or mental framework as to what would be possible for transfusion. So sometimes in transfusion, the thing that is given is not necessarily what is available, because if it's available and it is not compatible, then that becomes problematic. So what is normally being given is that which is suitable for the specific situation or the specific case. And we have to take into consideration the population expression as well, so as we can use that information of their race, the demographic information, to determine what is their medical condition, the disease state, to determine what is the most suitable and viable product. So as we continue our journey through the immunohematology aspect of the profession of medical technology, medical laboratory science, clinical laboratory science, laboratory medicine, I hope that this will give you insight as to some of the theoretical frameworks that are used in the different investigation in coming to a conclusion as to what is most suitable in different situation and cases. With that said, thank you once again for spending some quality time with us. I hope that the information that you gained was meaningful, was impactful, and you have a better grasp and understanding.
There's something that many persons fail to realize as it relates to the ABO blood group system in terms of the molecular genetics. Some persons believe that there is only one ABO allele, but since cloning was taking place in the 1990s, the complementary DNA corresponding to the messenger RNA transcribed at the blood group ABO loci, it was determined that more than 250, yes, you heard that right, there are more than 250 ABO alleles that have been identified by molecular investigation. And the ABO genes is located, as we had disclosed before, at chromosome 9. And in this area, it consists of 7 exons. The last two exons, the 6 and 7 exons, they encode for the catalyst or the catalytic dominance of the ABO glycosyl transferase. So exon 6 and 7 actually constitute 77% of the gene, which most of the coding sequence is lined in number 7, right? So amino acid substitution result primarily from the deletion, the mutation, or gene recombinations within these two exons of coding DNA. So from this, we can derive variant ABO glycosyl transferase, which are responsible for less effective transfer, or they react weaker when the conventional sera is used, anti-sera, anti-A, anti-B is used in identifying the blood group. The ones that we need to bear in mind, the 7 that we need to bear in mind is of the common ABO alleles includes the A1. Yes, A1. Remember, A1 is the one that we use in the reverse blood grouping. Then we have A1 variant, which is A subscript 1 and V. And with that in mind, let me just put that on the screen so that we can all have a better visual. So the 7, I'm going to put it there, 7, SEVEN, the 7 alleles that we need to be mindful of, all right? And these are the ABO alleles. So ALLELES, all right? So number 1, we're talking about the A1, and that is the A1. Then we have number 2, the A1V, which is the A1 variant. All right, and remember we said we have A1 and we have A2. So in your mind, I want you to think about having an A2. So you have A1, A2. So you're saying to yourself, is there an A3? Actually, there is an A3, but it's not a part of this the 7 allele. The next one, number 4, is going to be the B1. Then we have 5 is the O1. Yes, and 6, guess what? We have a variant again, so it's going to be O1V because it is the O1 variant. All right? So this is O1 variant. Then number 7 is the O2. So yes, we have A2 and we have O2. So this is just something for you to be able to mentally visualize that these 7 ABO alleles do exist. And as we will come to note, the A portion has
also many variants. But if you divulge a little bit into molecular genetics, which is a part of the whole systematic training in becoming a medical technologist, a laboratory medicine practitioner, a clinical laboratory scientist, medical laboratory scientist, whichever route you decide, then you will be given some idea as to how molecular genetics plays an important role in the ABO blood group system. So if we are focusing in that area, we can go a little deeper. But for this study, for this moment in time, we will leave that detail for another sitting. So I want you to understand that all ABO antigens arise from mutation in the single ABO gene. So only 3 specific mutations show a higher frequency in the population, indirectly leads to the change in the epitope structure, resulting in A, B, or O specificities. All right. So there are just 3 mutations that occur that has been noted to change your blood group. So somebody can be A and they present as B. All right. And somebody can be A and then change over to B. So when this happens, this is determined to be an incompatibility issue that is taking place, and then we as a clinical laboratory practitioners have to decide how do we go about resolving these anomalies, these discrepancies in the ABO. So the two mutation of substitution changes the specificity of the enzyme for the alpha N acetylgalactosamine transferase. All right, and this is the enzyme for the A, or the A enzyme. All right, and then it would convert from that to an alpha 3D galactosyl transferase, which is the B enzyme. The A more difficult to call, the B a little easier to call. All right. So these glycosyl transferase in turn introduce an alpha 1,3 N acetylamine, which is the A, or it can also introduce alpha 1,3 galactose, which is the B carbohydrate at the end of the H oligosaccharide or the glycan chain, right? The third mutation can be a deletion within the 5' region. And you know that when we're doing molecular genetics, we have the prime region, we have the folding, we have the replication process of the DNA and the RNA taking place. So this is one of the 5' location or region that a mutation can occur, and this mutation at the 5' is usually a deletion. So a deletion within the 5' region of the catalytic dominant creates a premature stop to the codon that inactivates the enzymes altogether, leaving the H glycan unmodified. So you have H that are unmodified, right? So these different glycans differentiate the A, the B, and the O antigen or the epitope. So when we have this situation, we realize that we can have discrepancies in our interpretation of the blood group system. Now, Let us try to wrap our minds around the formation of the A, B, and H soluble antigens. So we already discussed the A, B, and H antigen as a whole. Now let us talk in more detail about the formation. So the ABH antigens, these are integral part of the cell membrane, especially the red blood cell, the endothelial cell, platelets, lymphocytes, and the epithelial cell, just to name a few. The ABH soluble antigen can also be found in all body secretion. And you may be aware that we spoke about the SE, which means secretor, and then we spoke about when we talk about secretion, we're thinking about things that are displayed in other areas. All right, so there are different areas in which secretion occurs from the body. You have the sweat, you have the saliva, you have the All right, we're gonna get into that in a little while. So I just want you to just wrap your mind around that idea that your ABO blood group system, in part, can be identified based on secretions. All right, so let us go back now to the ABH soluble antigen. So the ABH soluble antigen can also be found in all body secretion, right? Just remember that. So their presence is dependent though on the ABO genes inherited and on the inheritance of another set of gene called the SE, which is the secretor genes. And these genes regulate their formation. So even though we have the ABO gene, we need to have the secretor gene activated so that we can have the secretion being easily identifiable in our body fluids. All right, which are easily accessible to the majority. All right, beautiful. So we are getting a clear understanding. What we should note that 8% of random US population are known as secretors because they have inherited the capital S, lowercase e, capital S, lowercase e, or capital S, lowercase e, lowercase s, lowercase e. All right, let me just put that secretor genes. Comes to us from the SE S, let me, SE SE. All right, so this is the genes that is necessary in order to be a secretor. You can all either have SE, capital E, common, capital S, common E. Then we have capital S, common E. So with this, you have the secretor. Number two, you can also have the secretor with capital S, lowercase e, lowercase s, lowercase e. All right. So the inheritance of the SE gene, we spoke about this, the FU T2. This here is the SE gene is coded for. So the inheritance of the FUT2, which is the secretor genes, codes for the production of transferase. And the transferase that we're talking about is the alpha 2L. and it is fructosyl transferase. F U C O S Y L T R A N S F E R A S E. All right, and that is used as a modifier. It modifies the type 1 precursor in the secretion of the H substance, and we know that H substance is necessary to determine your ABO ABH blood group system. All right, it can then be further modified to express A and B substances in the secretion. And when we think about secretion, the first thing that's supposed to come to our mind is saliva. Because in the mouth we have amylase, we have secretors having their blood group easily being determined just by a buccal secretion assessment. All right, so we are thinking along those lines. So a group A individual who is a secretor will secrete glycoprotein carrying the A and H antigen. So if you are a group A, you will carry, based on your secretion protocol, so SESE or SESE, you will secrete the glycoprotein and this glycoprotein that you will secrete carries the A and the H antigen. So now you can see why you use anti-A, anti-B, anti-H in our determination of blood grouping system. All right, so that is just something for us to think about. So but what if the person is B? Let's go to that. So the person is group B. We're going to expect them to have the secretor genes as well, SESE and SESE, right? And they're going to still have their glycoprotein and then they are going to be giving us a direction towards the secretion of the B and the H anti gene. All right, it is the presence of the SE gene specified let's say alpha 2L fructosyl transferase that determines whether an ABH soluble substance will be secreted. So people who inherit the SE, we need to talk about this. People who inherit the SE, these are lowercase So persons who inherit the SE genotype are termed non-secretor. So if you inherit those genes, you are a non-secretor. And that is just how it is. So some persons are secretors, other persons are not secretors. So what I want us to take this time to do is to just go over some of the differences between the ABH antigen on the red blood cell and the ABH soluble solution or soluble substances. All right, so let us gear our minds. Let me try to do a division on this board so that we can all see. So we're going to talk about the ABH antigen on RBC. All right, and then we're also going to be talking about the ABH soluble SOLUBLE substances. All right, so the ABH antigen on the red blood cell. First thing we need to remember is that the red blood cell antigen can be a glycolipid, it can be a glycoprotein, it can be a glycophospholipid. All right, so we have three, 1, 2, 3. So it can be a glycoprotein, glycoprotein, glycolipid, glycolipid. It can be a glycosph sphingolipid, lipid. All right, but for the secretion, the ABH secreted soluble or ABH soluble substances, secreted substances are only glycoprotein. All right, glycoprotein. So they are only glycoproteins. So that is one distinction that we have already. So the next thing that we want to remember is that for the antigen, the red blood cell antigen are synthesized only on type 2 precursor chain. So synthesize SYNTHESIZ on only type 2 precursor chain. All right, the next thing, if that is on type 2 and we talk about type 1, I want you to put in your mind, if this is type 2, the other one must be type 1. All right, so for the AB and H soluble substances, secreted substances are primarily synthesized on type 1 precursor chain. All right, so synthesize SYNTHESized on type 1 precursor chain. All right, now let us think about it. We had discussed this already, that the type 2 chain refers to the beta 1, which has 4 linkages, in which the number 1 carbon of the galactose is attached to the number 4 carbon of the N-acetyl glucosamine sugar of the precursor substance. All right, sorry about that. All right, so we have type 2, and it's the same thing that we have been discussing before. So type 2 goes to the beta 1, and this is linked to 4. It has 4 linkage, LINKAG, right? That's what we need to understand. So 1 is linked to 4. Now, for the type 1, which is on the AB and H soluble substances, we have type TYPE1 chain, all right, and then, as we stated, referred to as the beta 1, and that is linked to number 3, or it is 3 linkage. All right. So for the A, B, and H, we have 1 to 3. And the 3 is a linkage in which the number 1 carbon on the galactose is attached to the number 3 carbon on the N-acetyl glucosamine sugar of the precursor substance. All right. Now, let's move right back to the ABH antigen on the red blood cell. What I want you to bear in mind is that the enzyme produced by the H is FUT1. The gene is alpha-2L fructosyl transferase, which acts primarily on type 2 chain, which are prevalent on the red blood cell membrane. All right, so remember the enzyme. So when we talk about ABH antigen, we're talking about enzyme ANZYME produced by H is FUT1. All right, just remember that. And for the AB and H soluble substances, the enzyme produced by the secretors, so enzyme produced by secretors, and the secretors are the SE, and we know that this SE is going to be FUT2. All right, that is a gene, and this gene is still, we're still talking about the alpha-2L fructosyl transferase. All right, its preference is towards action on the type 1 chain in the secretory tissue. So if we bear that in mind, we are on good terms in understanding the concepts of these ABO system, not to mention the ABH antigens on the red blood cell. Then we have the ABH soluble substances, which when we talk about soluble substance, we're talking about the substances that are secreted. So this here gives us an idea that the ABO blood group system is not as basic as many persons may consider it to be, but it has some complexity within it. However, it is the basis on which the modern blood transfusion system is based. So let us just talk about in brief the idea that ABO has subgroups. And the original report of most ABO subgroups were made before the availability of the monoclonal typing reagent currently used in routine ABO grouping. So the ABO subgroup represents the phenotypes showing weaker and variable serological reaction within the commonly used human polyclonal anti-A and anti-B, not to mention, and anti-AB reagents. So some labs use A and B, only, while other labs use A, B, and AB. All right. So the AB is usually used to confirm the A or the B and to find out if there are any variants in the whole system. All right. So, what I want us to do in our next sitting is to go into some details about the A subgroup, because yes, we have A1, we spoke about that. We have A2, we spoke about that. We mentioned A3, but there are other A's that we could investigate. And I want you to be on the journey as we investigate those A subgroups. But before we delve into that, I want you to remember secretion and the secreted genes, SE, and also the A, B, and H substances. All right? So when we're talking about body and body fluid, and we're thinking about the ABH substances, I want you to understand that they can be detected in saliva, they can be detected in tears when you cry, they can be detected in your urine when you urinate, they can be detected in the digestive tract when the food is being digested, they can be detected in the bile coming from the bile duct, they can be detected in the milk because the milk in lactation for mammals is a secretion. They can be detected in the amniotic fluid, which is the area in which house the infant. They can be detected in pathological fluids such as the pleural fluid, the peritoneal fluid, the pericardial fluid. It can be also detected in ovarian cysts.
The ABO blood group has what we consider to be subgroups. Basically, they are groups within a group. That is basically the idea of a subgroup. The original report of most ABO subgroups were made before the availability, as we discussed previously, of the monoclonal typing reagents, all right, which are currently used in routine ABO blood grouping, which is one of the most common testing procedures that are done in the blood banking laboratory. So the ABO subgroup represent the phenotype showing weaker and variable serological reactions with the commonly used anti-A and anti-B and anti-AB reagents that are currently being used in our laboratories today. But the subgroup that I want us to begin our Cavalier's investigation concerning is the A subgroup. Yes, remember we spoke about A1, we spoke about A2, and we made an allusion to A3. But guess what? There are more subgroups of A. So it was in 1911 when von Dungerms described two different A antigen based on reaction between A red blood cell and anti-A and anti-A1 lectin. So the A1 is the one that is predominantly observed in our population, and that is why we use the A1 lectin to take from the A people to get those who are A1, and we pool those together, and then we have our antiserum. But this A1 lectin is derived actually from a plant base, which we'll get into in a little while. All right. So group A red blood cell that react with the anti-A and the anti-A1 are classified as A1, right? Because they react with A, anti-A,
And usually if you react with A, it means that you have the antigens on your surface of your red blood cell. And if you have the A and then we react to you with the A1 lectin and you give a positive result, which means that there is some agglutination taking place, it really indicates to us that you are positive for A1, which means that you have the A1 version of the A. All right? So that is just a simplified version of the whole process of approach, right? So if a person doesn't have the A1, what do they have? So whereas those who react with anti A and not with anti A1, these people are usually classified as A2. Yes, A2. So we know A1, now we have been exposed to A2. So the red blood cell from A1 and A2 individuals reacts basically equally strong. So yes, there is a reaction that is observable, and we can use this to determine the blood type of the individual. So they react with the current reagent monoclonal anti-A in the forward grouping. So in the forward grouping, because they have the A antigens on the cells, it can be identified with the use of the monoclonal anti-A, which is in uh, the vial, and it comes in a blue color. All right. So the A subgroups are more common than B subgroups. So yes, we are alluding to the fact that A subgroup exists and we also have B subgroups that exist. So the A subgroups are more common. And we have seen both A and B in practice. The weaker serological reaction of the ABO subgroup is attributed to the decreased number of A and B antigen sites on the individual's red blood cell. So, depending on how many antigen binding sites you have on your red blood cell, that would give an idea as to the reactivity and the expression that will be observed, right? So, the classification of the A into A1 and A2 phenotype accounts for 99% of all A individuals. So if you know the A1 and you know the A2, guess what? You know 99% of what you need to know about the A people. All right. So the cells of approximately 80% of A or AB individuals are A1, or if they're AB, it could be A1B. All right, because with AB you have A and B being expressed. All right, or if it is the A person, it is just A1. Now, the remaining 20% are A2, or for the AB person, it would be A2B. All right, so understanding this based on the results that we get from our typing and the use of the A1 lectin, we can determine if a person is A1 or A2. If the A1 and the A2 are not differentiated, then we'll just put A or AB, right? But if we know, we use the A1 lectin and then we get a positive reaction and the person is A, we know that this person is A1. If we use the A1 lectin and we get a positive reaction with the AB person, we know that this person is A1B. All right. And if it is not positive when we use the A1 lectin, then we know that this person is A2. And if it's an AB person, then the person is going to be A2B. All right. And these tend to react a little bit weaker. So the difference between the A1 and the A2 are both quantitative and qualitative. So quantity, when we talk about quantitative, and quality in terms of how they interact. All right, so just bear that in mind. So if we look at the quantitative and qualitative differences of A1 and A2, we can look at the number of antigen sites, the amount of transferase enzymes, the amount of branching that is taking place. For qualitative, we can talk about the difference in the precursor oligosaccharide chain. We can talk about the subtle difference in the transferase enzymes. We can also talk about the formation of the anti-A1 in in a percentage of some subgroups. All right, so these are just some things that we should bear in mind as we continue our investigation on the A1 and A2. So we are talking and we are working our way around to a complete understanding. But in order for us to grasp everything, we need to remember that the production of both types of antigen is a result of the inheritance of the genes on the ABO. So A1 individuals or A2 individuals, the gene is inherited on the ABO locus, right? So inheritance of the A1 gene elicit the production of a high concentration of enzyme alpha 3 N acetyl galactosamyl transferase. All right, remember we said that the A's are harder to pronounce, so yes, this is the A again, which converts almost all the H precursor to A1 substances. All right, on the red blood cell we're talking about. So they are very potent in terms of their expression. They created about 810,000 and 1,170,000 antigen sites in adults with the A1 red blood cell, whereas the inheritance of the A2 gene result in a lower production, in which we have what we consider to be maybe 240,000 to approximately 290,000 antigen sites on the adult A2 red blood cell. So this is something that is very interesting, and it gives us an idea as to the epitope and to the antigen binding sites that are available. And based on the availability, we can determine what is the expected reaction that we can perceive. All right. So the A2 allele is characterized by a single base substitution at the nucleotide 467 and a single base deletion at the nucleotide 1060, and we normally refer to it as 1060 DEL deletion of this nucleotide. All right, so this is what we normally refer to it as. And remember, most of this reaction takes place in 6 and 7, but in this situation, it is taking place in exon number 7. All right, so let's just do a quick recap again for the antigen binding sites. So For the A1, which we made mention of, it has 810,000 to 1,170,000, right? Then the A2 has less antigen binding sites. They have 240,000 to about 290,000, all right? And this is in adults, in adult ADULT humans. So we have a nice understanding of what is taking place. And let us not forget the sugar. So it's alpha 3 N acetyl ACETYL galactosamine nil transferase, TRANSFERASE. All right, beautiful. So now we have that documented, and we have a visual representation imprinted in our mind. All right. So these substitution alters the activity of the site specificity. And when that happens, there is a challenge in the coding region, and the A glycosyltransferase is diminished in some instance. The immunodominant sugar on both the A1 and the A2 red blood cell is the N-acetyl D-galactosamine. All right. So the dominant sugar, let me just DOMINANT sugar for the A1 and the A2 is N acetyl CETYLE D, don't forget the D, the N, the D, and gal lactose amine. All right. So now we have that. So the qualitative differences that exist, because there are quantitative and there are qualitative difference. So since 1 to 8% of the A2 inherited individual produce anti-A1 in their serum, and 22 to about 35 of the A2B individuals produce anti-A1. So even though these individuals are A2 majority, they still have some level of A1 that exists. So this gives us a little idea as to the technicalities that are about to unfold. So this antibody can cause discrepancies between the forward and the reverse blood grouping. So when we're doing the blood typing, we may get one reaction in the forward and we say, yes, this person looks like an A, and then when we check out the reverse is saying that this person is O, because there's no reaction, or there is a problem in the interpretation. In the forward, we may get A, and in the reverse, we may get that, hey, this person may be B, and then that there is problematic. So there are many different kind of discrepancies that can occur, all right? But we have to be mindful to know what to expect, and when we know what to expect, then we can move into performing the relevant procedures in order to get that which is expected, and to determine why we are not getting what is expected. So this system of the A1 and the A2 subgroup is very intense, but it is something that every student should know and understand. So the anti-A1 is usually naturally occurring, which means that, as we stated before, when we are born, we already have our antigen binding site. They just mature a little bit more when we come to maturity, 2 to 4 years old. All right. So what we need to remember is that these antis, they are IgM. So the anti-A1 is an IgM, cold reacting antibody, and is unlikely to cause a transfusion reaction because the body is usually at about 37 degrees when blood is being transfused. So if you have a cold reacting and your body is already at 37 degree, then the likelihood of a transfusion reaction from the activation of that antibody is negligible. All right. So let us bear in mind that the body is at 37 degrees. We normally transfuse blood at 37 degrees. So the blood is warm and it is transfused. So the antibodies that will be considered to be clinically significant in terms of can mount or evoke a transfusion reaction would be those that react at 37 degrees. All right. So just bear that in mind so that you can navigate your notes and have a better understanding and appreciation for what is happening. So the antigen presence on the red blood cell of the A1 and the A2 individual can be depicted in two ways. And this depiction, most often A1s are illustrated as having both A and A1 antigen on the cell surface, in contrast to the A2 cells, which only contains the A1, the A rather. All right. So the A2 cell only contains the A. Alternatively, the A1 red blood cell can also be conceptualized as having only A1 antigen sites and the A2 also only having A antigen sites. But in routine forward grouping, reagent anti-A strongly agglutinates both A1 and the A2 phenotype. Differentiation of the A1 and the A2 phenotype is determined serologically using the A1 lectin. And this A1 lectin, this is very important for us to know. The A1 lectin is made from a seed of a plant. It is called the Dolichos bifloros. All right, let me write that down because I have had many students who have seen this question come on the ASCP examination. All right, they ask about the A1 lectin and its derivative, where it is derived, and it is from a seed. All right, so seed, that's a seed, SEED. All right, that's where it comes from, and the name of the plant is Dolichos, Dolichos bifloros. So it's Biflorus. All right, and this is all italicized. All right, so this is a plant. So let us just bear that in mind. So the lectins are seed extracts that agglutinate human cells with some degree of specificity. The A1 lectin agglutinates A1 or A1B cells, but does not agglutinate A2 or A2B. So one is for the A group and one is for those who are AB. All right? So we have that all clear. Due to the A2 glycosyl transferase being less efficient at adding the immunodominant sugar to the H antigen precursor, the A2 red blood cell shows an increased reactivity with the anti-H lectin. All right, and this is very important for us to know. So we have what we call the anti-H lectin. So anti-H lectin, and this has a very special name as well. I want you to remember this name. It's called ULEX, ULEX, and Euro, PAEUS, ULEX europaeus. All right, that's how we normally pronounce it, but if you have a different pronunciation, please use it. But the idea is that you know how to identify this name and its association. All right, so Ulex europaeus. All right, and this here is the anti-H lectin. So H antigen is found in a greater concentration on the RBC of group O individuals, right? Because they did not differentiate to produce A or B, so they have just the H. And in the ABO blood grouping, we use anti-A, anti-B, anti-AB for the forward grouping. We don't use anti-H unless we're doing some investigation. So if we have an O individual, we are expecting that there will be a predominance of H antigen that is found on that individual. So the H antigen may not be detectable in the A1 individual because of the presence of the A1 gene, which almost all of the H antigen is converted to A1 antigen. And this conversion takes place by a sugar reaction. All right. So the H antigen is converted to A1 antigen by placing the large N-acetyl D-galactosamine sugar on the H substance. All right. So due to the presence of so many A1 antigen, the H antigen on the A1 and the A1B red blood cell may be hidden and therefore may not be available to react with anti-H antisera. All right, because if there's none present, or if all of them have been used up, or if the titer is too low based on the volume that is available, then you can expect a reaction to be almost non-existent. All right, so just bear that in mind and bear the concepts of how we come to the understanding in mind as well. So in the presence of the A2 gene, only some of the H antigen is converted to A antigen, and the remaining H antigen is detectable on the cell. So if you use the anti-H with the A2, you will be able to find or get a stronger reaction with the anti-H than you would if you did it with the A1 individuals. So weak subgroups of A antigens will often have an inverse relationship between the amount of H antigen on the red blood cell and the amount of A antigen formed. The H antigen on the A1 and the A1B individuals is so well hidden by the N-acetyl D-galactosamine that the anti-H is occasionally found in the serum. This anti-H, guess what, is naturally occurring. So we are thinking about IgM cold agglutinin that reacts best at room temperature. As can be expected, this antibody is formed in response to a natural substance and reacts most strongly with cells of group O individual. So just bear that in mind as you navigate the concepts of the A and A subgroups. All right. So let us create a mental picture, a mental representation of the information that we have just received. All right. So we have established that we have two variants in terms of the phenotype of A. So A subgroup, let me just write it, subgroup, Group. All right. We have the blood group, and then we have the anti-A reagent, because we're going to react anti-A reagent with it, anti-A reagent, which is yes, and then we're going to use anti-A1 lectin, anti-A1 lectin. All right, so a A1 person is going to have a reaction with the A. Let me all right. So the A1 person is going to have a reaction with the A and also a reaction with the A1 lectin. An A2 individual is going to have a reaction with the A, but they will not have a reaction with the A1 lectin, because the A1 lectin is specific for A group with the A1. All right. So that is all you need to have an appreciation for at this time. All right. So let us try to do a reaction to determine the A substances. So if the blood group is A1, what antigen? Let's do, this is a blood group, and then we're going to do the antigens present, and then its reaction with the antisera, which is the anti-A, and anti-A1 lectin. A1. So this is the A1 lectin. Bam, bam, bam. All right. So this person is A1. So if they're A1, we expect them to have A1 and we expect them to have A. All right. They're supposed to have a reaction with the A as is, and they're supposed to have a reaction with the A1 lectin. If they're A2, we're expecting them to have A, but they won't have the A1. And we're expecting them to react with anti-A, and we are not expecting any reaction with the A1 lectin. All right? So that is the simple essence of the reaction profile that is expected for an individual with A1 or A2. All right? Beautiful. So now let us move in a little further to finish our investigation. Let us try to do a forward and a reverse blood grouping, or yes, I think we have time. We could do a little forward and reverse blood grouping. So the person is A1. This is a phenotype. Let's do the phenotype. Phenotype. The anti-A. So that's anti-A. And this is anti-B. And this is anti-AB. Right? And anti-A1. So we're just going to do this section first, and then we're going to move down and add. Let's do the A cells and the B cells. All right. So if a person is A1, we're going to expect a reaction in the A, no reaction in the B. We're going to expect a reaction in the B, AB rather, and we're going to expect a reaction in the A1, right? So if you have A antigen, you're not going to have A antibodies, you're going to have B antibodies. So no reaction and 4+ reaction, all right? So that is just basically what we're expecting. So the person is A2. We are going to also expect a 4+ reaction. We're going to expect a reaction in the A. We're not going to expect anything in the B. In the AB, we're going to expect something. Anti-A1 lectin, there's not going to be any. A cells, there's not going to be a reaction. Then we're expecting there to be a reaction in the B, maybe 3+, 4+. It all depends on the individual, all right? So that there is for us to have a appreciation of. So we recall that there are different antigen binding sites. So the A1 would have about 810,000. This is antigen binding site, and 1,170,000 approximately, all right? And the A2 would have about 240,000 to 290,000, right? So this is just for us to wrap our minds around the information. So A1 and A2, there is substance that is present in the saliva, and let me just put it right here, substance present in saliva. In saliva, and that would be the A and the H. So that is in A1, and the same thing in A2. So just keep that in mind, all right? So we were discussing previously that there is a difference in the reactivity of the anti-H antiseria or the anti-H lectin with the ABO, all right? So I want you to bear this in mind. The O individual has the most amount of H, all right? So O has the greatest amount of H, greatest amount of H. And the A1B individuals has the least amount of H. So this H, right? So this is the most, and this is the least. So A2 individuals have less than the O, right? But the B individuals have less than A2. Guess what? The A2B individuals have less than the B. And the A1 individual have less than the A2B. And the A1B has less than the A1 in terms of the presence of H. All right? So just bear this in mind so as you can know what to expect with your different investigations that you may undergo in terms of studies to determine what is present and what is absent, right? So now let us try to work our minds around a few more subgroups. So there are different subgroups. We call them weak A subgroups. And we did mention some like the A3, that's a weak one. We have AX, we have AN, we have the AM, we have AY, and we have AL. All right. So we're going to get into them maybe in another sitting right before we touch the weak B subgroups.
In the last sitting, we discussed the A subgroups. In specifics, we spoke about the A1 subgroup and the A2 subgroup. We discussed how we could identify and distinguish the A1 versus the A2. We had come to the conclusion that if we add anti A1 lectin to known A cells, then if the result is positive or there's an agglutination, that individual would be A1. If the result is negative, then that individual would be A2. And if the person is AB and the same principle is applied to the A portion and the A portion indicates to be positive, then that person is A1B. And if the A portion is negative in the AB person, that individual is A2B. Right? So we have that all cleared in our mind, and we also established that there are more antigen binding sites on the A1, 810,000 to about 1,170,000 approximately, while on the A2 we have a lower number of antigen binding sites, about 240,000 to 290,000 on the red blood cell site. All right, so if we have that in mind, we can understand how the degree of reactivity will be when we add the various antiseria. And the A1 and the A2 exist, but there are other subgroups that exist. We're going to try to touch a few of them so that we can have a better appreciation. But before we delve any further, I want to bring our attention to some structural characteristics of the A1 and the A2 RBC. So let us put that on the board. Structural characteristics. All right, so the A2, let's start with the A2. The A2 red blood cell predominantly has A, and AB. All right, so let's put that. Possesses A superscript A and A superscript B. All right, and this causes a situation where there is the unconversion of the H3 and the H4 antigen site.
So unconverted, like when you have unconverted Christians, persons professing to be Christians, but they are not truly Christians in the way that their religion indicates that they should be. So these are unconverted. They should be expressing the H so that they can be A1, but they are not expressing it, so they are A2. All right, now let's move now to the A1. So in the A1 red blood cell, we can identify, this is what is very important, we can identify the A superscript A, plus we can identify the A superscript B, plus we can identify the A superscript C, plus we can identify the A superscript D. So if you do the A, B, C, and D, right? And in this situation, we know that there are no unconverted H3 and H4 antigen site. And because there are no unconverted site, all of the sites are representing the A1. All right? So that is just something that we need to understand. So that is a structural characteristic that I think would be necessary for us to appreciate a little bit more as we progress. Now, we have been speaking about the possibility of a A3. And yes, we have A3 available for us to investigate. And let us talk about it. The characteristics of the weak ABO phenotype. So let us put it up. Of weak ABO phenotype. And we're talking about the A type, right? Or let's call it a subgroup. So let us say, what phenotype are we looking at? So let's write the phenotype, and then we're going to try to see the reactivity that would exist between the anti-C or the anti-A, the anti-B, anti-AB, and the anti-H, right? And then we're going to see what is present in the serum of this individual. Now, the phenotype, then we're going to look at the reagents, reagent. So the phenotype, we want to start with A3, then we're going to look at the anti-seras, anti-A, then we're going to have anti-B, anti-AB, and anti-H. So 1, 2, 3, 4. All right. So we can see what is happening. And then in the serum, so let us put a big double line right here and a big double line right here as we move over into the antibodies in the serum. All right, so we're going to think about having the anti-A, we're going to think about having the anti-B and the anti-A1. Let me see if we can get this a little bit better. Let me see if we can structure this a little bit better so that we don't have much confusion. So let me go into the pen. Let's bam, bam, and then we're gonna have bam, bam. All right. Let's carry it across. Then For this, we're going to have anti-A, anti-B, anti-AB, and anti-H. Then in the serum, we want to know if there's anti-A, we want to know if there's anti-B, and if there's anti-A1. All right? So, and then we're going to have the phenotype as usual. So now let us start with A3. So for the A3 person, there may be some form of mixed field reaction with the anti-A. So you may get a 2+ mixed field reaction. For the anti-B, there is no B present. For the AB, the same reaction you get in the A, you tend to get in the AB portion. And with the anti-H, remember with the A3 subgroup, this one has H present for all of it is not fully converted, right? So this here is going to give us a 3+ reaction. When we talk about in the serum, we're going to have no anti-A's, but we're going to have anti-B. With the A1, it may or may not react. So let us put plus or minus, so that is sometimes, all right? So sometimes it will react, other times it will not react. Now, let us look at the AX, because we have an AX subtype or phenotype. And with this one, you will get a weak reaction or no reaction taking place with the anti-A. When it comes on to the anti-B, you're going to have no reaction because it's an A individual, not a B individual. For the AB, you're going to have a 2+ reaction. But when it comes on to the H, there's going to be so much H there. You're going to have a 4+ reaction. So you're going to have a field day with the H's, right? In circulation, you may or may not have anti-A. And if it is present, it's going to be weak. For the anti-B, you probably have a little bit present, yes. And A1, there is a high possibility that you're going to have that present, all right? So this here, always you're going to have the A1 present. Then let us move to another one, A and E and D, all right? And this is not the one that is the end of the world, but this is very important as well. So for this one, we have a weak reaction in the anti-A, and we may have a mixed field, a weak mixed field reaction in the anti-A. For the anti-B, we're not going to expect anything because it's a patient that is A and not a patient that is B. And the same reaction we have in the anti-A, we expect that reaction to be in the AB. So the weak mixed field reaction. And this one, everything is not converted. So we're going to have a 4+ reaction. When it comes on to the serum, what is present in the serum, we're not going to have any anti-A in the serum, but we will have the anti-B in circulation. And when it comes on to reacting with the A1 lectin, we will sometimes have a reaction. So we're going to say plus or minus, right? Now, let us try another one. There is a special kind called AM. Yes, AM. So for this one, the anti-A, we have no reaction, or it's a weak reaction in the anti-A. When it comes on to the anti-B, we're not going to expect any reaction. So none of the A is supposed to react with the B, right? Because if it's just A, you're not going to react with the B any at all. All right? And for the anti-AB, we may have no reaction or a weak reaction, maybe a 1+ reaction. And for the anti-H, everything is not converted, so you're going to have a 4+ reaction taking place. When it comes on to the antibodies in the serum, we are not going to have any anti-A, but we are going to have anti-B. And guess what? It's not going to react with the A1 lectin. Then, after we move past that, we're going to have AY, yes, and we're not talking about the AY service that they have at church. We're talking about the A lowest subscript Y, all right? So for the anti-A, you're not going to have any reaction whatsoever. And this is something that I want you to bear in mind. And for the anti-B, there's no reaction. For the anti-AB, guess what? No reaction. But when it comes on to the anti-H, there's a 4 plus reaction. Now, in circulation, we're not going to have any A antibodies in circulation, in the serum, but we're going to have the B in the serum, and there's going to be no reaction with the anti-A1. All right? Now, there is another A subgroup that I want us to bear in mind, and that is the AL, like Elohim, but it's not Elohim, it's A subscript EL. All right. So for the anti-A, anti-B, and anti-AB, there is no reaction. It's similar to the AY, the church situation. But when it comes on to the anti-H, there is a 4 plus reaction. When we move now, this is where we get things a little bit tricky. When we move now into the antibodies, when it comes on to the anti-A being present, sometimes, so we're going to put plus or minus, we may have anti-A in the serum. So plus or minus. Anti-B, we are going to find the anti-B, and guess what? The A1, it is going to react with the A1. So positive, 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 positive reaction you're going to expect with the A1. All right? So these are just some characteristics of the weak ABO phenotype that I think that you should bear in mind so that you can have an appreciation for the whole design of the ABO blood group system. All right? What I want us to look at now is to see if we can determine if the substance is present in saliva of the secretor, or what substance is going to be present in those secretors or those with the secretor genes. All right? And to what degree is there a present, whether it is there's no present, there's a little trace, there is present, there is rarely present, or there's sometimes a present. So let us go into characteristics, same characteristics of the weak ABO phenotype. And we are talking about A's, right? A subgroup. Right, so let us make our table, phenotype. We're starting from scratch again. And then, if the person has a secretor gene, we want to know the substance that is present in the saliva. So substance present in the saliva. And we want to know the presence of the transferase in the serum, all right? So let us put presence of transferase in serum. All right, beautiful. So let us make our table and get ready to rumble. So we discussed the subgroup. So now in your mind, you should know the subgroup. We have A1, BAM. That is what majority of the population have. We have A2, BAM. We know that already. Then after that, we have some other subgroup. We have the A3, good. We have the AX. We have the AN. We have the M, we have the AY, and then we have the AL, all right? So all of those are subgroups, and we just need to bear them in mind. So let us start with the A3. So the A3 subgroup, we are expecting the presence of A and H. A and H, all right? And we know that there's going to be H present because the anti-H reacted at 3 plus, right? And we know that there's going to be A present because there was a mixed field reaction with the A. And not only that, it was a positive mixed field reaction. And transferase in the serum, we may have this sometimes. So this is not all the time, but sometimes we may have it. Now let us move into AX. And we're not talking about your X, we're talking about AX, the blood group, the subtype, all right? So substance that is present in the saliva, we are looking at the H. And the reason we are looking at the H is because anti-H was 4 plus, but for the anti-A, we realize that there is a weak or no reaction taking place. So majority of the time, you're going to have the H. So the presence of the transferase is going to be rarely. All right. Now for A end, E and D, and all of these are subscripts. For A end, we are going to have the H present. All right. The H is going to be present. And the transferase in the serum is not going to be there. So there's going to be no transferase in the serum. For the AM, we are going to have both the A and the H. The anti-B had a slight positivity. The H, anti-H, had a 4 plus positivity. And yes, there is going to be transferase present. Now, for the AY, we are going to have the AH, and there's going to be trace amounts of transferase. And now the AL, Elohim, AL, we are going to just have the H present, and there's going to be no presence of a trace transferase in the serum. All right, so just bear those in mind as you go on your journey to explore the different classes of the A blood group, the ABO blood group system, and understand that these phenotypes do exist. So when we observe these phenotypes in our practice, there is a possibility that these will be implicated in a discrepancy, and these discrepancies have to be solved. Most times in solving these discrepancies, what has to be done is an enzymatic technique or an elution technique in order to determine the weaker subgroup and to distinguish them, all right? So serological techniques are usually used in order to distinguish and classify these subgroups. It is very important for us to just have an appreciation that even though we are accustomed to someone being a person can be A something else. It can be A1, A2, A3, AN, AX, AM, AY, AL. It all depends, all right? So I want us to have a mental picture as to a flow chart. So the cell reaction. So the red cell reaction with the anti-A and the anti-B AB. So if there's a weakly agglutination taking place, the possibilities that we're looking at are A3, AX, AN, right? If there's no agglutination taking place, we're looking at absorption and elution of anti-A. So we're thinking about the possibility of AM, AY, and AL. All right, so let us try to put that on the board. So red blood cell reaction with anti-A and anti-AB. All right. So if we have a weak agglutination, weak agglutinates, we are thinking about possibly A3, we're thinking about AX, and we're thinking about AN. All right, good. And just peradventure, there is no agglutination. We have to examine a little further and do some absorption. ABSOR and elution of the anti-A. All right. So when we think about this, we are thinking about the possibilities of AM. We're thinking about AY. And because we're already in church, let's just go to AL. All right. So this is just something that I want you to bear in mind. So now, if we get a mixed field reaction when we do this, so we get a mixed field reaction, we are thinking of A3. If 10% of the cells show very weak mixed field, we're thinking AX. All right. If we have weak agglutination with anti-AB only, we're thinking about AN. All right. So that there is just an idea for us to proceed with our investigation. So let us move into the non-agglutinations. All right, so when there is anti-A that is easily absorbed and eluded, and secretors demonstrate quantitatively in the saliva, we are thinking about AM. When the antibody absorption and elution occur, and the secretors contains a small amount of A in the saliva, we're thinking about AY. When the A absorption and elution occurs, and secretors contain only H substances and no A substances in the saliva, we're thinking about AL. All right, so bear that in mind. So for AM, let's put it down. AM, we do the anti-A elution. This is easy elution, EASY elution. All right, and we have secretion of A substance in the saliva. All right, now for the AY, we have anti-A eluded. All right, and then we have small amount, just small amount of A substances in the saliva. All right, and then the big boy. We have anti-A absorbed and eluted. So anti-A absorbed plus eluted. But this is the kicker. We have only, so let us write that big and bold, ONLY, H substances. H substances. We are thinking about an A that is L. All right? So we have only H substances and no A substance in saliva. All right? So just like that, we have covered majority of the A subgroups that we need to know. But we discussed that we have A subgroups and we also have B subgroups. So yes, we are going to have to go through the B subgroups. And when we talk about the B subgroups, I want you to think about having B3, just like how we have A3, we have BX, we have BM, we have BL, and we have the regular B, as the phenotype, the regular phenotype. So I just want you to digest this information. And in the next sitting, we're going to try to examine and explore the B subgroups. And then after we have done that, we're going to try to move our attention to just understanding in detail a little bit more about the Bombay phenotype.
In the same way we have the A subgroup, we have the weak B subgroup. So a subgroup of B are very rare. So we have more likelihood of finding an A subgroup than a B subgroup. All right. So as we were saying, the subgroup of B are very rare and are much less frequent than the A subgroup. You see that subgroup B are usually recognized by a variation in reaction strength using the anti-B and the anti-AB. Inheritance of B subgroup, similar to that of a majority of the A subgroup, is consequently or is considered the result of the alteration of a allele at the B loci. So for the A, it's at the A loci. For the B, it's at the B loci. Criteria used for the differentiation of the weak B phenomena include the following technique. So we're going to have to look at the strength and type of the agglutination with the anti-serous that are used, usually the ones that we are giving our eye focus on are anti-A and anti-AB. And in that mix, we also use the anti-H. Now we also look at the presence or the absence of the ABO isoagglutinins in the serum. So we need to know if we have isoagglutinins that are present in the patient's serum. So that's give us an idea to where to look. We need to investigate by absorption and elution studies with the B. So the presence of B substances in saliva gives us an indicator, and you must
be thinking about this by now. We can use molecular testing to determine the B subgroup. Now, red blood cell demonstrates serological activity for the B subgroup people that are weaker than normal designated B phenotype. All right. So we have the B3, we have the BX, we have the BM, we have the BL phenotype. There are no B subgroup reported that are equivalent to the A and the AY in the A subgroup category. So a classification system similar to the A subgroup has been used because of the common serological characteristic. So you may remember what we stated for the A subgroups. We're going to use a similar classification for the B subgroups, all right? So serological techniques can be used to characterize a B subgroup into the following category. We have B3, BX, BM, BL. Those are the ones that we're going to be focusing on. The characteristics of each weak subgroup are represented by the following, and we're going to get into those in a little while. All right. So the B3 phenotype generally results from an inheritance of a rare gene at the ABO loci and is characterized by the mixed field pattern of agglutination with the anti-B and the anti-AB. Then the BX, we have the BX red blood cell is typically demonstrated and is observed with the weak agglutination of anti-A, not anti-A because this is a B, so it's anti-B and anti-AB. And then we have other variations, the BM. So the BM red blood cells are characteristically unagglutinated by anti-B and anti-AB. But the BM red blood cell easily absorb and eludes the anti-B. All right. So let us just bear that in mind so that we can have a better appreciation as we move forward. But did we mention the BL? In the BL, red blood cells are unagglutinated by the anti-B or the anti-AB antisera. All right. This is extremely rare. So this is a rare phenotype and must be determined by absorption and elution of anti-B. So no glycosyl transferase has been identified in the serum of serum or RBC membrane of the BL individuals. So BL is inherited as a unique mutation in the exon 7 of the B gene at the ABO locus. Simple. Now, let us get into creating our flowchart so that we can have a better appreciation for the B phenotype. So, characteristics. Let's put it. So, characteristics of B phenotype. So, we're going to list the phenotype. So we have the phenotype. The next thing we want to do is to determine which reagents will react with this phenotype. Then we want to know about the antibodies in the serum. All right, so let's bring it down. For the reaction of the reagents, we want to think about the anti-A, anti-B, anti-AB, and anti-H. All right. Then we want to determine if this reaction is common and unexpected. All right. Now let's start with the B. A regular B individual. We're expecting no reaction in the A. We're expecting a 4 plus reaction or a strong reaction in the B, a strong reaction in the AB. And for the anti-H, we're expecting about a 2 plus reaction in the anti-H. And when it comes on to antibodies in the serum, we are going to be expecting there to be anti-A. All right. Not only that, when it comes on to unexpected, this one we're going to select none. All right. Now, that is the regular B. So this can be our framework as to what is a normal reaction to expect. Now, let us move to the B3. So with the B3, we're going to have no reaction with the A, anti-A, but when it comes on to the B, we're going to have a plus positive reaction, but it's going to be mixed field usually. And then for the AB, we're going to have a positive reaction and it's going to be mixed field, the same kind of reaction that we got in the B. And for the anti-H, we're going to have a 3 plus. In circulation, we're going to have anti-A, and we're not going to consider any to be unexpected. So that there concludes that section. So the only difference is the mixed field reaction, the positive mixed field reaction in anti-A and anti-B, and a higher grade reaction in anti-H. As we will realize with the subgroups, they have a high grade dead reaction with the anti-H. Now, let's go to BX. So when we talk about BX, XXX, we're talking about something that is weak. So there's no reaction taking place in the anti-A, but there's a weak reaction that happens with the anti-B and an equally weak reaction that happens with the anti-AB. And we expect to get a 3 plus reaction approximately with the anti-H. In the circulation, we are going to expect to find anti-A. And unexpected, guess what? We are going to have weak anti-B. So unexpected, this is very important, weak anti-B. So for the BX, we just want to remember that it's weak for the anti-A, anti-B, and we have a 3 plus reaction, the anti-H. And because this reaction is weak, we're going to expect some weak, unexpected anti-B to be present in the serum. And what is commonly found is the anti-A. So now let us move to BM. So with the BM, we can have a situation in which, similar to the others, there is no reaction in the anti-A, but as it comes on to the anti-B and the anti-AB, we're going to expect no reaction or a weak reaction to take place. No reaction or a weak reaction. But with the anti-H, we're expecting, as with the other subgroups, a 3 plus reaction. Then things kind of go back to normal. So in the serum, we have anti-A, and we have no none for the unexpected antibodies in circulation. All right, beautiful. Now let us go to BL. So with BL, this one is a little bit tricky in that for the anti-A, there is no reaction. For the anti-B, no reaction. For the anti-AB, there is still no reaction. But when it comes on to the anti-H, we have a 3 plus reaction. All right. When we check the serum antibodies, we are expecting to find anti-A. And when it comes on to unexpected, sometimes we can have a weak anti-B. So sometimes weak anti-B. All right. So this is for us to have a understanding as to the whole matrix of the B phenotype system and their characteristics. So I want you to just remember the operation. And from this operation, then we're going to deduce what are the possible outcomes based on the reactions that we get. So when you're in the real world, you will have a ledger or you'll have a key to guide you as to the interpretations of the various reactions. With that key, you're going to follow the guide, and that will get you to the answers that you so seek to determine about the patient different phenotypes. All right. So now let us do the presence of substance in the saliva, because you know that if we have a secreted gene, there is a possibility that there is antibodies present in saliva. And then we're going to look at the presence of the B transferase in the serum. All right. So those are two things that we're going to look at. We're going to look at the phenotype. Then we're going to look at the substance present in saliva. And then the presence of B transferase in serum. So let's get to it. So for the B person, the substance that we are expecting to find are the B and H. All right. And you'll realize that for all of these subgroups, there is going to be the H, because with the anti-H, we had some form of a reaction. The B had straight B had the weakest reaction, which was a 2 plus, and the others had a 3 plus reaction. So all of them we're expecting to find at least the H. So just bear that in mind, all right? And I just want you to wrap your mind around it. The ones that reacted weakly with anti-B and AB, or didn't have a reaction with anti-A and anti-B, those will usually only have H. All right? So just bear that in mind as we proceed. So for the regular B, you have B and H that can be present in the saliva. And when it comes on to B transferase, yes, there is the presence of B transferase in the serum. All right. Now, when we go to B3, we are going to have B and H because we had observed a mixed field plus positive mixed field reaction with anti-A and the anti-AB. All right. So we have that B being present. And sometimes, yes, we will have the B transferase and we'll get a weak positive based on that mixed field that we had, right? Now, we move to BX. So with BX, we had a weak reaction in the anti-B and a weak reaction in the anti-AB. In this situation, we're going to have H present because anti-H was 3 plus. But there is going to be no B in the saliva. And there's going to be no B transferase, all right? So if there's no B in the saliva, no B transferase is observed for this one. Now, for BM, we had a situation where there is no to a weak reaction. So in this case, we will have B, and there is a possibility of H, right? Yes, there is going to be B transferase, and this is going to be a weak positive as well, just like the B3, right? And the final one we're going to look at is the BL. With the BL, we are going to observe something similar to the BX because the BL had no reaction with the anti-B and no reaction with the anti-AB. So what is going to happen is that we had a 3 plus reaction with the anti-H. So in the saliva, we can identify anti-H, but because of the nil or the non-reactivity with the other antis that could be identified in the saliva, we will have no B being secreted. And if there's no B being secreted, then there will be no B transferase in the serum. So there is a no. So this is just something that I want you to bear in mind as we continue to examine the different subgroups of the ABO blood group system. So A's have their subgroups. B also has their subgroup. And when we understand these subgroups, it will give us guide as to what to expect when a discrepancy is observed. So understanding the existence of these subgroups and how we can navigate to determine what subgroup it is, when we get the conflicting reaction of the forward and the reverse, then we are in a better position to deduce and determine what is causing the anomaly. And when we know what is causing the anomaly, then we can move into operation to determine how do we resolve this anomaly. Because we have to remember that before we can dispense any blood or blood products, we have to determine what the patient's blood group is and give group specific. If there is a situation where the blood group cannot be determined without the implicated markers, we have to hold off on a transfusion that is not an emergency. But if it's an emergency, then you know that the routine blood banking procedure would indicate that we give the patient the O negative blood. But depending on the age of the patient, we will give them the group specific blood. So these are just some things that I want you to bear in mind as you continue to peruse the concepts as we seek to understand in whole the ABO blood group system.
As we have been discussing, we have a phenotype called the Bombay phenotype. And the Bombay phenotype was first reported by Bende in 1952. That name is pronounced or spelled rather Bhende. And in 1952, the location of the discovery was Bombay, India, or Bombay, India. So the phenotype results from the inheritance of a double dose of the H genes. All right. Remember, we said that in order to be O, you have to have H, capital H, lowercase h, or capital H, capital H. That way you can have an expression of O. But in the Bombay situation, you have lowercase h and lowercase h gene being expressed. So the production of Bombay is very rare, but it does exist, and it's something that we need to be mindful of. So this phenotype, Bombay phenotype, can be represented genotypically by the lowercase h. As a result, the ABO genes cannot be expressed and the ABH antigen cannot be formed since there is no H made in the Bombay phenotype. So because there is no capital H, and you know that we need the ABH system to be in effect so that we can have the production of A people, B people, or O people. So the H helps us to determine what the blood group is. And the H is the one that directs the body in producing the various antigen markers. But in relation to the Bombay individual, there is no capital H. And if there is no capital H, there can be no A, there can be no B, and the person cannot be expressing an O phenotype. It may look like O, but it is not O. And we will come to understand exactly what is happening with this phenotype. So in the Indian state of Andhra, we would realize that there is a prevalence of this phenotype. And when we say prevalence, we are talking about in comparison to the average population. So the Bombay phenotype is about 0.048%, and that there is considered a prevalence. All right. In comparison to in Europe, the Bombay phenotype is 0.0003%. So we are seeing how rare this phenotype is. I want us to wrap our minds around this whole phenomena of the Bombay phenotype. We had discussed previously that if you see something looking like, let's bring it up on the screen, O, and we have the lowercase h, this is not an O person. This is the representation of the Bombay, Bombay. This is the Bombay phenotype.
FEPHENOTYPE. All right, and the genotype is denoted by HH. So this is the Bombay genotype. So if you see this or you see this, we are talking about the Bombay blood group. Now, in understanding the underlying molecular defect that occurs in the Bombay blood group, we have to realize that this is not a normal occurrence. And if it's not a normal occurrence, then we are going to be looking at the possibility that this could be considered a mutation. And indeed, it is a mutation. And a mutation in the FUT1 genes, which is the genes that codes for the H. FUT1, this is the H genes, and this is where we have the mutation. So the Bombay mutation occurs in the FUT1 genes. And when this mutation occurs, what happens is that there is the production of the genes being silenced, which means that this gene is now incapable of coding for the enzyme alpha 2L fructosyl transferase, which is the H transferase. All right, so we have a mutation. So we have the Bombay blood group. This is the phenotype, which equals the genotype. All right, and this occurs due to a defect in the FUT1, which is the H genes. All right, when this defect occurs, what happens is that there is the silencing of the gene. Of the gene expression. All right, and then when this happens, we realize that the H transferase is inhibited. So the enzyme that is responsible is for the H2L FUCOSYL TRANSFERASE. All right, and this is the H transferase. So this is basically an idea of the overview of what is happening. So as we proceed with our investigation on this phenomena, we need to understand that when this happens, the enzyme that normally catalyzes the transfer of the L-fructose in an alpha 1,2 linkage to the terminal on the galactose of the precursor molecule on the RBC forming the H, that there is inhibited. So the mutation, this mutation that we're talking about, the Bombay mutation, underlines this kind of expression. And even though this mutation affects the FUT1 genes, the H genes, it also affects the FUT2 genes. And the FUT genes is the SE genes, which is the secretor genes, all right? And these genes code usually very similar to the alpha 2L fructosyl transferase. That when it is activated, it transfers fructose to form the H antigen in the secretion. So this genes is usually responsible for the expression of your blood group in your body fluid, like your saliva, your intestinal tract, your amniotic fluid. So this is the secretor genes that allows this to occur. All right. So the FU1 genes, FUT1 genes is silenced, and nobody likes to be silent, but this gene is silent, and this is the H genes. So if there is no H genes being expressed, then we can expect that there is going to be no A, no B, and no H antigens being observed. And then we have another challenge. The FUT genes is also inhibited, in which this genes is the secretor genes, the SE genes. And this genes is usually what allows our blood type to be detected in our body fluid. So now that we have an understanding of that phenomena, we can go a little deeper into observing some more intricacies as to this rare situation. It is rare, but it does exist. And if you're in the Indian population, then there's a probability that you may know of someone that has this phenotype. When family studies demonstrates the ABO genes is inherited in the Bombay phenotype, the genes are usually written like this. So you know that we have normally, so normal, we have A, we have B, and we have O people, and we have AB individuals. All right? But when the gene study for the Bombay, we normally write it like OHA. So that's Bombay A. All right? OHB. That's Bombay B. Or OH, and these are lowercase h's. So OHAB. So this is how it is normally represented. So since these A red blood cells lack the ABH antigen, they fail to react with the antisera. The anti-A, it's not going to react because there are no A antigen binding sites on the red blood cell. The anti-B will not react because there are no B antigen binding sites on the red blood cell. The anti-H will not react because there is no H present because the H genes has been silenced. All right. So in RBC testing using the anti-A and the anti-B, the Bombay would phenotypically looks like a O person. So let us put it on it. You have anti-A, you have anti-B, and we have anti-H. All right, so let us put the group, GROUP. This is just a forward grouping, forward group. We're not going to use the AB, but let us put the AB just so that we can help someone who may needs to understand it fully. Anti-AB and anti-H. So the anti-H is what we are focusing on, and then this is the phenotypic group, FE. Let's just put the blood group. All right, so this person is O. If a person is O, you're going to expect no reaction with the A, no reaction with the B, because there's no A or B antigens. There's no A or B antigens, so there is no AB reaction either. But you're going to expect a reaction with the H, right? So this person is going to be O for the forward. For the Bombay, so this here is the Bombay. Let me circle that. So it's going to look similar to the O. So no reaction with the anti-A, no reaction with the anti-B, and there's no A nor B, so there's going to be no reaction with the AB. And for the anti-H, this genes has been silenced, so all of this silence. So it's going to look like a group O, but this is not a group O because of the anti-H inability to respond. It gives us an indication that something is wrong. And this is just in the forward grouping. So this here gives us an idea as to how we'd go about investigating a discrepancy if observed. So this is just something that you need to bear in mind, so that if you ever come across it, maybe not in real life, but maybe in an examination, you'll be able to dissect to understand what is happening and how you would go about resolving this situation. So the Bombay serum contains anti-A, anti-B, anti-AB, and anti-H. And we're going to have to understand it. If it doesn't have the anti-H on the cell, we're going to expect it to be in the serum. If it doesn't have the anti-B, it's going to have it in the serum. If it doesn't have the anti-A on the anti-A antigens on the red blood cell, it doesn't have the B antigens on the red blood cell, it doesn't have the H antigen on the red blood cell, we're going to expect, because they're not present on the red blood cell, we're going to expect that they're going to be in circulation. And when they're in circulation, they can then be identified. So, in the serum, of the Bombay individual, we can find anti-A, we can find anti-B, we can find anti-H. So we need to bear this in mind, right? Unlike the anti-H found occasionally in the serum of the A1 and the A1B individuals, the Bombay anti-H can often be potent. And guess what? This is what is very important. This anti-H reacts strongly. So this anti-H is potent. It reacts strongly at 37 degrees Celsius. It is an IgM antibody capable of binding complement. So it's capable of binding complements. It's like complement binding potential. And if this occurs, it's going to lead to red blood cell RBC lysis. So you understand how dangerous this is. Transfusing normal group O blood with the highest level of anti-H concentration, the H antigen concentration, to a Bombay recipient with the H in the serum would cause immediate cell lysis. For the Bombay individual, the only blood group that can be transfused to this person is from another Bombay representative. All right, or let's call it recipient. So the ABH substances is also absent in the saliva because it messed up the FUT2 genes. So you cannot use the saliva or the body fluids to determine the exactness of the blood group. So let us go over some general characteristics again of the Bombay phenotype. So the Bombay phenotype, this is going to be important for us to note, especially if a question comes up or if we are at a reference laboratory in Mumbai, India, or in one of the areas in which there is prevalence of the Bombay blood group. All right. So for the Bombay blood group, we know that it is represented genotypically as HH lowercase. All right. So this is the genotype, GENOTYPE. And we know that there is no H antigens formed. No H antigens are formed because it silenced the FUT1 genes, which is the H genes. All right. And if there is no H antigen formed, then we know that there is no A or B antigens formed. The reason we know this is because the H genes codes for the A and the B, right? We know that phenotypically when we do the forward grouping, it looks like O, but it's not an O. All right, so it looks like O, but it's not O. In the serum, we will find whatever is not present on the antigen or the surface of the cell as antigens we're going to find those in the serum. So we know that there's going to be anti-A. We know we're going to have anti-B. We know we're going to have anti-H. So all of these are present in the serum. All right. For this individual, even though we normally say that if you're a blood group O, then you are a universal donor, especially if you're O negative. In this situation, this is an exceptional situation. This is an exception to that rule. All right. So for the Bombay individual, they can only receive transfusion from an individual with the same blood group, or let us put from a Bombay individual, Bombay, or Bombay individual, which is denoted by OH. All right, so we have that in the back of our mind. So as we move forward, we are in a state where we can better appreciate the arrangement of this blood group. So I want you to also bear in mind some general characteristics of the Bombay blood group. We discussed them prior. So the general characteristics: we have the Bombay, B-O-M-B-A-Y, Bombay, and it is represented by O, capital O, and subscript, and then it is represented by the H, null, because there are no H phenotype. All right, so we know that there is absence of the H, the A, and the B antigens. So absence of the A, H, and B antigens. Hence, there is no reaction with the AB or the H lectin. All right? And if it's absent on the surface of the cell, it's going to be present in the serum. So in the serum, we're going to have anti-A, anti-H, and anti-B. Not only that, the H here is going to be very, very potent. All right? Now, the ABH does not secrete because this mutation impacts or impairs the FUT2 genes. All right? So no secretion. Is observed, and we're talking about secretion of the A, the B, and the H. All right, so you will not find this in the saliva. So when you do the saliva test, there will be no blood group determined from the saliva, because the substances that are being looked at will not be observable. Very good. So we're having a field day. Now let us continue. We indicated that there is an absence of the alpha 2L fructosyl transferase, which is the H enzyme. So there's absence of the H enzymes. And if the H enzymes is absent, then we know that we won't be having any coding for the A, the B, or the H. But there is a presence of the A or B enzymes in the serum depending on the genotype. So there can be the presence of A or B enzymes in the serum based on the genotype. And this is the ABO genotype that we're talking about. All right? A recessive mode of inheritance has to be indicated. And with this recessive state, what happens is this. The individual presents with HH. So it's lowercase h, lowercase h. The red blood cell of the Bombay phenotype will not react with the anti-H, which is classically known as the Ulex europaeus. All right? The red blood cell of the Bombay phenotype are capable only, are compatible rather, only with serum from another Bombay individual. So if we give O to a Bombay, then that there is instant termination of the said individual. So we have to bear this in mind as we understand the complexities surrounding the various blood groups and the different anomalies that may be observed as we investigate rare cases that exist. And the Bombay is one such case. It was because of the Bombay blood group why I was fascinated with immunohematology. And another reason for my fascination with immunohematology was the idea of the chimera, genetic chimeras. I read about it and I just fell in love with the whole phenomenon. And from then, it led me to wrap my mind around immunohematology, blood banking, transfusion medicine, whatever you call it in your jurisdiction. And since then, I have spent over 10 years in practice of transfusion medicine. So I am excited for you on your journey as you seek to develop and understand, appreciate the concept, so that's when you are deployed into the work world or while you're exercising your duty, you'll be able to make critical, logical decisions which will safeguard the safety and ensure the efficacy of the blood and blood products that are transfused.
The ABH antigen and antibodies have a role to play as it relates to a disease individual or a diseased state. So the associations between the ABH antigens are practically in any disease can create complications. Some persons have many myths that they say one blood group is better than another blood group, that one group of person like the A blood group is more inclined to criminal activity, and B blood group uh, may easily have hangovers. Persons with O blood group may have good teeth. So all these things are not a true reflection of the blood grouping system, and they are just myths. So bear that in mind when you hear someone is saying that if you're this blood group, then this will happen. That is just fabrication, and you have to be wary and cognizant of that. But what I can say though is that various disease states seems to alter the red blood cell antigen. And when there is an alteration from normal functionality, what results is either progressively weaker reaction or the addition of an acquired pseudo antigen, which can be seen during either the forward, where we use the anti-series blood grouping. So when we use the anti-A, the anti-B, the anti-AB, the anti-D, and sometimes depending on what kind of grouping methodologies we're doing and what investigation, when we use the anti-H, we will realize that there is some weak reaction that is not really what we expect to see, but that is what we're getting. The next thing we want to understand is that persons with the big C, a lot of persons have the big C. First, it was the big A, now it is the big C. When we talk about cancers, there is a proliferation of a growth of a particular cell, and this can impair the blood grouping system and its representation. So when we think about a leukemic individual, a leukemic patient, a patient with leukemia, we have to think about leukemia, the chromosome 9 translocation, and hematologic disease that can induce stress. Let us think about hematopoiesis. So we're going to be running through our mind like thalassemia have been shown to actually depress antigen strength. So if you have thalassemia, there is a possibility that the antigen strength, which means that antigens are on the surface of the red blood cell, those will be weaker. So cells will appear to give a mixed field agglutination. When people have a disease we call the Hodgkin's disease, there has also been reports of a weakened or a depressed ABH red blood cell antigen, resulting in a variable reaction during the forward grouping, similar to those found in leukemias. So the reactivity when you have Hodgkin's disease, you have leukemia, and you have certain kind of cancers, can reduce the reactivity. So the antigen strength increases again, though, when the person goes into remission.
So, yes, a disease means that the body is in a state of disease, which means that the normal functionality, the normal operation, the normal protocols are not being executed in the way that the body would desire for it to be executed. And as such, there are challenges in the body's ability to mount up an adequate response to pathogens, to uh, disease causing agents, to protect itself, and even to show who it really is. And when this happens, we have a situation where the body is weakened and compromised. We also have the isoagglutinins. So we have anti A, anti B, or anti AB. These also may be weak or absent in leukemia, demonstrating hypoglobemia, globulinemia. All right. So you have low level of gamma globulins in the anemic state. All right. That's basically what the word. So hypogamma globulinemia. All right. So when we think about this, we're thinking about chronic lymphocytic leukemia, so CLL. So you may come across this when we focus on hematology, then we will see this being played out, and then we can put the pieces together to have the bigger picture. So various lymphomas such as malignant non-Hodgkin's variety may yield weak isoagglutinins. And this isoagglutinin is owing to the moderate decrease in the gamma globulin fraction. All right. If someone is immunocompromised, they have a immunodeficient state, or they have immunodeficiency disease, such as congenital gamma globulinemia, they will yield weakened or absent isoglobulin, and they will probably in some instance have weak or absence in isoagglutinins. All right. So if this problem is suspected, a simple serum protein electrophoresis will confirm or rule out this condition. Did you know that individuals with intestinal obstruction who have carcinomas of the colon or the rectum or some other disease of the lower intestinal tract may have an increased permeability of the intestinal walls, which allow the passing of bacterial polysaccharide from E. coli. And usually the serotype that we're talking about is O86. All right. And this will pass into the patient's circulation system and result in what we call the acquired B phenomenon, especially in group A1 individuals. So the A1 individual can have an expression of a B based on the intestinal situation that is taking place. So when there is some form of carcinoma, there is some form of cancer in the lower intestinal region, this individual becomes compromised, especially by E. coli, which the body should naturally be able to pass out. All right. But you know that we have different classes of E. coli. We have the hemolytic E. coli, we have the regular E. coli, and this one that we're speaking about in this case is the serotype O86. All right. Now, the patient with group A red blood cell will, what they will do is absorb the B-like polysaccharides, which react with the human source anti-B. So the E. coli possesses protein molecules. These protein molecules can evoke a similar representation as that of the, and then it reacts with the human anti-B. So basically, that is all that is happening. All right, very good. So the lack of detectability of the ABO antigen can occur in patients with carcinoma of the stomach and or the pancreas. All right. But serum contain excess amount of blood group specific soluble substances that may neutralize the antiserum utilized in the forward grouping have been observed in some instances. So these are just some things for us to note, that different disease state can impair how the blood group is interpreted. And with that said, I want us to be reminded that all ABO discrepancies must be resolved prior to a transfusion or the release of blood for a patient. So now, with that said, understanding this, that there are situations where the blood group that we get is not the one that is expected, let us embark on assessing the ABO discrepancies. So when we talk about ABO discrepancies, these are unexpected reactions that are observed in the forward when we add the anti-A, the anti-B, the anti-AB, the anti-D. If we're going to put that in there, because we usually do that part of the reaction. We're not going to talk about the RH system just yet. So we're going to just leave it at the anti-A, the anti-B, the anti-AB for now. But in the real world, we also in the forward have the anti-D as well. All right. Now, for the reverse grouping, there can be discrepancies. So it can happen in the forward or it can happen in the reverse. And with the reverse, we use the A1 cells usually based on the population type and the population availability of that blood group, the predominant blood group that is in the population. Then we use A1, and we also use B. So the A and the B to confirm the forward. So that there would be the reverse. So those are the cells we use in the reverse, and we use the anti-series in the forward. So these unexpected reaction can occur in either the forward or the reverse. But what if we indicated to you that it can occur in the forward and the reverse simultaneously? That is a possibility. So these can be problem due to uh, the patient's health situation. This can be problems due to exposure to something. This can be problem due to inaccurate sample. It can be a lot of problems that cause these discrepancies. So as clinicians, we have to be mindful in resolving these discrepancies so that the right patient receive the right blood that is compatible, that will be viable, as efficacious as it can be. All right. So technical errors are one of the errors that we need to speak about. So technical errors can also cause the ABO discrepancy. Example, if blood samples and tubes are labeled in error, failure to add the reagents as needed, you know, we have cell suspensions are too heavy or too light, so we have prozone or postzone reaction. Then we have clerical errors in which incorrect results have been documented. We have a mix-up of the sample, so the wrong patient sample with the wrong label. Then we have mixed observation of hemolysis. So we missed that observation of the hemolysis. So we saw hemolysis taking place, but we never picked it up at the time when it was happening. So we failed to realize that something was wrong. So failure to add reagents, failure to add the sample. So we're doing the testing now, we forget to add the sample. So that there is a problem. Failure to follow up with the manufacturer's instruction, because from time to time the manufacturer instructions will be altered, will be changed based on the batch of reagent that they're sending and what in-house testing they're doing to validate their study. So they may use a different reagent that need to react slightly differently than what you know, but you are going in with your mind to say, hey, this is how it has been done for the last 5 years, so this is how it should be done. But changes do happen over time. Then we have a situation where the uncalibrated centrifuge can either centrifuge too fast or centrifuge too slow, which means that you can miss the reaction, or when it's spun too fast, the reaction forms, but then it is broken up. So over centrifugation or under centrifugation, that there can be a problem. And if the centrifuge is not calibrated to the appropriate speed, then we are going to have a challenge. So we want to get rid of all of these technical errors that exist. If the reagents are contaminated, bacteria is in them, then they will fail to react as how they were intended to react. And sometimes we warm our specimen. So when we warm them, we can induce or introduce hemolysis depend on how we warm them. When we warm some reagent, we inhibit some of the reactivity. So we have to be mindful to avoid warming unnecessarily. So all of these situations can create a challenge in the blood grouping determination. But when the discrepancy is encountered, all results must be recorded, but interpretation of the ABO blood type has to be delayed until a resolution can be determined. And the information is recorded so that you can go back to see if what you got in the morning is what you got in the afternoon. And when we talk about morning and afternoon, we're not talking about in the morning when you did it or in the afternoon. We are talking about what you did first and what you did after to see if there is consistency. Just like when we have a transfusion reaction, we have to perform all the procedures that we did prior to dispensing the blood and blood products, and then checking again compatibility just to be sure. And then we're going to also perform a DAT on the first sample, the second sample, to see if there is any form of hemolysis, to determine if there is any form of sensitization taking place. So all of these things have to be taken into account. So transfusion medicine is very tedious, and you have to be on your A game if you're going to work in transfusion medicine. Working in transfusion medicine, working in blood banking, immunohematology is like working in the emergency room. So you have to just always be as sharp as you can be, spot any deviations as quickly as possible, and take the remedial corrective actions that are needed to resolve such discrepancies. So we have discrepancies, we have encountered them, we need to have a resolution. But we have a situation where we have ABO discrepancies and they are divided into different groups. And we're going to go through the groups: groups 1, groups 2, groups 3, group 4. And then we can come to the idea as to which group discrepancy we would prefer. So in group 1 discrepancy, discrepancies are associated with unexpected reaction in the reverse grouping due to weakly reacting or missing antibodies. And this one is kind of easy to identify, because the forward is going to be give you a positive reaction, maybe 3 or 4 plus, and then the reverse is going to be lacking. So when that happens, you know that something is off, right? So these discrepancies are more common than those of the other group. So we had indicated that when a baby is born, we don't do the reverse grouping because the antibody titer is so low. So if that is the case, then we don't expect to see a reaction. So if a person unknowingly takes up a sample and run it as an adult, but even though it's a baby, they will have challenge with reading the complete reaction. But the patient history is what will give us guidance as to what needs to be done. So for newborns, ABO antibody production is not detectable until about 3 to 6 months of age. Elderly patient, after you have reached up in age, your antibody titer tends to decrease. It's just like a female. When she is 20 years old, she is fertile. When she is 50 years old, her fertility rate is decreased. When she is 90 years old, it's almost non-existent. All right, so the possibility of impregnating a 90-year-old woman, no matter how young you may be, is almost impractical. But if you are strategic, it may be possible for you. So, but let us get back to the meat of the matter. The idea is that elderly patient, the production of the antibodies is depressed. All right. So patients with leukemia, example, the chronic lymphocytic leukemia or lymphoma, or patients with malignancy, such as malignant lymphoma, demonstrate hypo, which means low, gamma, globulinemia or globulinemia. All right. So it's hypo, gamma, globulinemia. All right. So it's low level of basically globulin, gamma globulin, which is protein, and leads to some level of anemia, right? So it's blood situations. And we will get a little bit more into that in a future sitting. So patient using immunosuppressive drugs. So the drugs that we use play a critical role in how our body reacts. So if you're on immunosuppressants, guess what? Immunosuppressants doesn't only try to help your body to recover by itself. It basically pushes down all the defenses. It tries to impale, and then it allows the body to remount a reaction. So this happens, and when this happens, also you have a situation where you have hypogamma globulinemia taking place as well. Patients with congenital or acquired gamma globulinemia or immunodeficiency disease will have or express this kind of discrepancy. All right. So patients who have bone marrow replacement, hematopoietic progenitor stem cells, transplants, HPCs, they can develop situations in which their forward and their reverse are not corresponding. So patient whose existing ABO antibody may have been diluted by the plasma transfusion or exchange transfusion can also have challenges with the reverse. And guess what? Patients who have ABO subgroup can have a problem with the reverse grouping. So in order to resolve this discrepancy, forward and reverse testing does not match. First thing you're going to do is investigate if an error in the specimen collection and identification is suspected. So we're going to try to get out the collection criteria where they met. All right. So we're going to note the initial testing time, date it was performed on the patient. We're going to request a new sample if we're suspecting that there was a collection error. Then we're going to repeat the testing. If no discrepancy, then we're going to report the ABO group that we have identified. All right. Then, if the initial testing was performed using patient red blood cells suspended in serum or plasma, we're going to have to wash those. We're going to have to wash the patient red blood cell with saline and repeat the testing. If no discrepancies, then we'll report out the ABO blood group. But if there is some unexpected reaction between the forward and the reverse testing, we have to look up the patient's information, the age, the diagnosis, the medication, because medication plays a very important part, the transfusion, pregnancy history. Then we're going to determine whether the discrepancy is in the red blood cell or the serum, resulting in the observed weakened reactivity. All right. For example, a patient with serum problem will have weak reactions in the reverse grouping. Patient with red blood cell problems will have a weak reaction in the forward grouping. Persons, let's say, both serum and red blood cell problem exists when there's a weak reaction in both the forward and the reverse. So, if it is a situation where the patient has a serum problem, we are going to try to run antibody screen, run autocontrol, and run a saline replacement to rule out rolo. And if that works, then results can be dispatched. If a patient has red blood cell problem, then we have to do a DAT and we're going to run auto control. If it has both, the patient has both, we're going to have to wash patient's cells with warm saline, not just regular saline, but warm saline, and we're going to retest. We're going to run a DAT, we're going to run an auto control, we're going to run antibody screen, because we have to try to figure out what is causing the discrepancy. So these discrepancies can be resolved with the appropriate procedure. Now let us move into type 2 discrepancies. So type 2 discrepancies are associated with unexpected reaction in the forward grouping due to weakly reacting or missing antigen. So type 1 was the reverse blood grouping. Type 2 is the forward blood grouping. All right, so when we think about the type 2, we're thinking about subgroups of A or B may be present. You know that we have different classes of A, we have different classes of B. So this now would give us a problem in determining the patient's blood group when we're using a commercial antiserial. If the person has leukemia, this may yield a weakened A or B antigen. And Hodgkin's disease have been reported in some cases to mimic the depression of antigens found in the leukemia. We spoke about the acquired B phenomena, which shows a weak reaction with the anti-B antiserial and is most often associated with disease of the digestive tract. So if the person has cancer of the colon, then their blood group may be what we say altered or changed. So when we have situations like this, we have to try to resolve the discrepancy. So the agglutination of weakly reacting antigens with the reagent antiserial can be enhanced by incubating the test mixture at room temperature for up to 30 minutes to increase the association of the antibody with the red blood cell antigen. If the reaction is still negative, then we're going to move to the next step, which is to incubate the test mixture at 4 degrees for 15 to 30 minutes. So we're going to include group O and autologous cells and controls. All right. So red blood cell can also be pre-treated with enzyme and retested with reagent antiserial. The acquired B antigen arises when the bacterial enzyme modify the immunodominant blood group A sugar, which is the one that we say was difficult to call, the N-acetyl D-galactosamine, all right, into the D-galactosamine, which is sufficiently similar to the group B D-galactose to cause the cross-reactivity, or cause it to cross-react with the anti-B antiserial. This pseudo-B antigen is formed at the expense of the A1 antigen and disappears following the patient's recovery. So after the person would have been treated for their condition, whether it be the E. coli has been removed from their system, they will have their blood group coming back to what it needs to be. All right, so blood group reagents are usually monoclonal. So the monoclonal B, it strongly agglutinates cells with acquired B antigen. The pH of the reagent contains E S4 has been lowered, and consequently only those cells with the strongest example of acquired B antigen react with the antisera. So we can use this to determine what is causing the reaction. All right. So there are other methodologies that we can use in resolving the discrepancy. But let us move into understanding the type 3. We did type 1, which was the reverse, type 2, which is a forward. Now let us move to type 3. So type 2 is the forward, type 1 is the reverse, type 3. These discrepancies occur between forward and reverse grouping and are caused by protein or plasma anomalies or abnormalities or anomalies, right? And results in rouleau formation. So rouleau formation basically means the cells stacking on each other to give you an appearance macroscopically as agglutination. So we call this a pseudo agglutination. All right. So this situation occurs when there is elevated level of globulin from certain diseases states such as multiple myeloma, Waldenstrom or Waldenstrom's uh, macroglobulinemia, other plasma cell situations, and advanced stages of Hodgkin's lymphoma can cause this to happen as well. So with type 3 discrepancies, we can have elevated level of fibrinogen. Yes. So if there's an elevated level of fibrinogen, then we are thinking type 3 discrepancies. When we have plasma expanders, such as dextran, we can expect this to happen when we have Wharton jelly in the cord blood. That is why we usually wash the cord blood before we perform the ABO testing, because we want to get rid of the Wharton jelly. So these things can impact and give us a discrepancy that we don't really need to have in our reaction. So the common resolution ruler is basically the stacking of the erythrocytes, the red blood cells, going on each other that adhere and forms a coin-like fashion. It appears in a coin-like fashion, giving the appearance of an agglutination. So this can be observed on a microscopic level. When we look at it from a macroscopic, just using our eyes looking at it, it looks like an agglutination, but that is not the case. We have to take that under the microscope to see if it's truly an agglutination. So cell grouping can usually be accomplished by washing the patient red blood cells several times with saline, performing a saline replacement technique with what it does is that it frees the cells. In the case of rouleau formation, in the reverse type, all right? In this procedure, serum is removed and replaced by an equal amount or equal volume of saline. In true agglutination, red blood cell clump will still remain after the addition of saline. Rouleau can be a nuisance in the laboratory since it is an in vitro problem observed during laboratory testing. It is not an in vivo problem from the patient. So it's basically the testing procedure, the conditions are not suitable that causes the rouleau formation usually. So we have to be very mindful of roller formation, and this can clog your hematology analyzer. It can cause you to spend a longer time in the transfusion department than you need to in resolving discrepancies. So we have to be very mindful of the rollers that exist. But usually washing the cells would get rid of most of the jellies that may be present, the excess proteins, as well with the replacement therapy that there usually helps. All right. Now let us move into type 4 discrepancies. So these discrepancies occur between the forward and the reverse grouping are due to miscellaneous problems that have the following causes: cold reacting or cold reactive auto antibodies in the red blood cells are so heavily coated with the antibodies that they are spontaneously agglutinating independently, that there can be problematic. So without even adding the reagents, auto antibodies just jump on each other and start to agglutinate. When we have a situation of a circulating red blood cell of more than one ABO group due to probably a transfusion, a bone marrow transplant, a stem cell transplant, we can have discrepancies. When we have unexpected ABO isoagglutins, that can be a reason to have a discrepancy taking place. When we have unexpected non-ABO alloantibodies, that too can create some discrepancy. What we are going to do is just in brief, think about it. How would we solve a situation like this? So the possible resolution to a situation where we have cold reacting autoantibody is potentially cold reacting autoantibody causing the spontaneous agglutination. What could we do? It's cold. So if we add heat to it, then maybe, just maybe, this will resolve it. So to resolve this discrepancy, the patient red blood cells should be incubated at 37 degrees because that is the temperature that the body reacts. Ideally, for a short period of time, then wash with saline at 37 degrees as well. So we're going to keep everything warm. We don't want the temperature to fall, because if the temperature fall, it will auto agglutinate again, right? We're going to wash with 37 degrees saline. So the saline has to be warm. Wash with 37 degrees saline 3 times, and then we're going to repeat the typing. If this is not successful in resolving the forward typing, the patient red blood cell can be treated with 0.01 mole DTT to disperse the IgM related agglutinin. As for the serum, the reagent red blood cells and serum can be warmed to 37 degrees for 10 to 15 minutes. So you're gonna do a mixed tested. It's gonna be mixed and tested, and then it's gonna be read at 37 degrees. So there are many ways that the discrepancies can be resolved. So these are just some of the discrepancies that exist. I just wanted you to have an idea that discrepancies in the ABO do exist, and we're going to go through the flowchart of resolving these discrepancies in our next sitting. So this moment was just for us to have the idea of the existence of these discrepancies. So now that we have the idea of the existence of these discrepancies, the next thing that we are going to do is to move our minds into resolving them.
Find challenges in determining the forward and the reverse blood grouping if there is a discrepancy, there is an anomaly, there is a situation that the patient is undergoing which is interfering with the reactions that are expected, are interfering with the reactions of the antigens, are interfering with the reactions of the antibodies. These exist, and we just have to be mindful of them. As it relates to type 1 discrepancies, I was almost going to say type 1 diabetes, but it is actually discrepancies. We're not talking about diabetes in this sitting. We may speak about diabetes when we're doing the chemistry review. But as it relates to the group 1 discrepancies, as I had alluded to prior, that the pivotal changing point of me electing to become a laboratory medicine practitioner was the concept of the chimeras. So, in the type 1 discrepancies, we have a system that exists, we have a situation that exists that we call chimerism, right? And it is just defined in brief as the presence of two cell population in a single individual. So you have one person with two different cell populations. So instead of having A or B or O or AB, you have a combination of two population. So that there is basically what is considered to be a chimera situation. And this chimerism was discovered in twins born to a group O mother and a group B father. So usually if a person is O, we're expecting them to have OO. And for B, we're expecting them to have BB or BO. So in that situation, the person can be O and the person can be, that's the offspring we're talking about, and the offspring can also be B because the O can come from the mother and one O can come from the father if the father is BO. And that there would give you the O. The B would be derived from a BO father giving the B and receiving the O. So when that happens, the child, the offspring, would be B. Or if the father is BB, then you will still pass on only the B and take on the O, and that would produce a B child. So that is basically what is the expected uh, result that we would foresee for a combination of this nature. So, as we were saying, that this chimera situation was discovered in twins born to a group O mother and a group B father with a mixture of both the B and the O cells instead of the expected B or O being expressed. Based on our combination, we understand what is taking place. So instead of having a blood group of A, B, O, or AB, this individual has a blood group expressing both B and O. So detecting a separate cell population can be easy or difficult depending
on what percentage of the cell is in the minor population and what percentage is in the major population, and what is present. So the reactions in these situations of chimeras are typically mixed field. Not because you see a mixed field reaction means that this person is a chimera. I want you to bear that in mind. But when we talk about chimeras, we have true chimeras, which only occurs in twins and is rarely found. So what happens is that two cell population will exist throughout the life of the individual. In utero, exchange of blood occurs because of vascular anastomosis. As a result, two cell populations emerges, both of which are recognized as cells. So the body is recognizing both groups as itself, which means that the body will not mount a reaction against itself in a normal functioning individual. So this is a concept in immunology. If the body decides to mount a reaction against itself, then we would have a situation which would be considered an anomaly. And this situation, you can think about it and then indicate to us below so we know that you know the answer. And as a result, we have two populations taking place in the chimera. All right. And the individual, remember, will have these two populations existing in their bodies from birth until non-existence. And the individual will not make the anti-A or the anti-B. So in this situation, we are expecting isoagglutinins to be present, but guess what? They are not present in the reverse grouping. And it depends on the percentage of the population of red blood cell that exists in each of the twins. If the patient or the donor has a history of a twin, then the chimeras may be due to the patient has a history of twins, then it may be a chimera. If the patient has no history, then and they express a chimerism, what we could conclude is that this may be due to dispermy, which is basically two sperm fertilizing one egg. So we have two sperm, two sperm, fertilize one egg. All right. And individuals that is created is of a chimera expression, if we can call it that. And this would indicate mosaicism. If we it's called mosaicisms, something to that effect. So let me just spell it for you. It's mosaicism. All right. So that can exist. More commonly, artificial chimeras occur, which use a mixed cell population as a result. And these can be found in situations where there is blood transfusion. So let us say, for instance, group O cells are given to a or a B patient. So a chimera temporarily can emerge based on the reaction. Then in situation where we have transplanted bone marrow or hematopoietic progenitor cell stem cells, that's HPCs, of a different ABO, we can have the development of a chimera. When we have exchange transfusion or fetal maternal hemorrhage, this can also induce a situation where we have chimerism being expressed. So we just need to be mindful of these situations that exist, and as we traverse our career in laboratory medicine, medical technology, clinical laboratory science, being a medical technologist, whatever it is, if you are practicing on the bench, you will be at the front line of this situation, providing that you're in an area where it occurs. And remember, even if you're in an area where it occurs, this situation is so rare that it is seldomly identified, but it is a possibility. All right. So just be mindful of that. When it comes on to type 1 discrepancy, we can have a situation in which we have a patient with a blood group. Let's do our forward grouping, FORWORD, forward grouping, and reverse group, REVERSE group. All right. So the forward group, we're going to have the anti-A and just the anti-B. Those are the only ones we're going to be using right now. And we may have the A1 cells and the B cells in the reverse grouping. A patient, let's say patient X, gives us a reaction of no reaction in the forward, no reaction in the anti-A, no reaction in the anti-B. When it comes on to the A1 cells, there's no reaction, and when it comes on to the B cells, there's no reaction. This here is problematic, and usually a type 1 discrepancy is considered if there is a challenge in the reverse blood grouping, which means the A cells and the B cells are overreacting or non-reactive. So in this case, if this is a type 1 discrepancy, we are assuming that there is something wrong in the reverse. The antigens on the cells are usually reactin 3 and 4 plus. All right, so this would generally give us a 3 or 4 plus. If it's not reacting, then there's a high probability that the forward is usually the correct group. So in this situation, we are going to assume that this may have been a baby. This could have been a baby. This could have been a geriatric patient, an elderly patient. So those things do occur. All right. Beautiful. So now let us look at our chimera. So let us say that we have a patient. We're going to call them twin 1 and twin 2. And we're going to do the forward, FORWARD. Then we're going to do the reverse, REVERSE. So we're going to use the anti-A, anti-B. Let's put the anti-AB as well, anti-AB. And for the reverse, we're going to do the A1 cells and we're going to do the B cells, all right? And then we're going to determine the blood group based on what we see. So, patient comes in, nothing in A, nothing in A for both twins. All right, that's fine. Then for the anti-B, we have a 2+ mixed field reaction. All right, mixed field reaction. For the AB, we have a 2+ mixed field reaction. Okay, all right. For the A cells, we have a 4+ reaction. Okay, and for the reverse, we have 0. That's for twin 1. For twin 2, nothing taking place in the anti-A. In the anti-B, we have a weak reaction. In the AB, we have a similar weak reaction. And for the A cell line, the anti-A1 cell reactivity, we have a 4+ reaction and no reaction in the B. All right. So in this situation, the 0 denotes negative, the MF denotes mixed field, and the WK denotes a weak reaction. So what we are observing here seems simple, but this is the red blood cell percentage, RBC percentage, that is being expressed. So for twin 1, we have 70% of B and we have 30% of O. All right. For twin 2, we have 30% of B and 70% of O. Now, isn't that amazing? That there is just an example of chimera situation that may be observed. All right. So now, let us bring our attention a little bit to the group 2 discrepancy. And the group 2 discrepancies are usually the discrepancies where we have challenges in the reactivities in the forward blood grouping. All right. So we have weakly reacting or missing reaction in the red blood cell grouping, which may be due to an excess amount of blood group specific soluble, or the BGSS substances present in the plasma, which sometimes occur with certain disease. So the disease state that an individual has predisposes them to produce certain or evoke certain reaction, such as when they have carcinomas of the stomach and the pancreas. We have seen excess amount of BGSS substances to neutralize the reagent anti-A and the anti-B, leaving no unbound antibodies to react with the patient's actual cells. And this will then yield a false negative or a weak reaction in the forward blood grouping. So the normal procedure to get rid of this is to wash the patient's cells of the BGSS substances with saline, and this should alleviate the problem, which will allow the forward grouping to correspond and correlate with the reverse grouping as expected. There are situations where antibody to low prevalence antigens in the reagent anti-A or anti-B can also cause a weakly reactive or missing reaction on the red blood cell when the blood grouping is done. But I must let you know that it is not feasible for the manufacturer to have all the blood group antigens to a particular in a particular anti-seria, because that would be taxing, time consuming, and cost prohibitive. All right, so we have to just bear that in mind. So when we have these discrepancies, we just try to use what we know to determine what we don't know, and then from that we deduce what is in the best interest of moving forward with the patient's blood group. So even though the situation is rare, it has been reported that the additional antibodies in reagent antiseries have reacted with corresponding low prevalence antigen present on the patient's red blood cell. Yes, everybody has a different mix of antigens on their red blood cell based on your genetics, based on your culturalization, based on your exposure. All these things can create different expressions of antigen binding sites. But there are some that are high prevalence, some that are low prevalence. Those that are low prevalence are the ones that sometimes are implicated in this kind of discrepancy. The procedure is, as we stated before, to get rid of it is to do washing, right? In situations like this produces an unexpected reaction of the patient's cell with the anti-A and the anti-B, or sometimes both, depend on the blood group, and it mimics the presence of weak antigen. So just bear that in mind. And the best way to resolve the discrepancy is by washing and repeating the forward typing using anti-sera from a different lot number. That usually just helps you to alleviate all the problems, all the challenges that you have. All right. So don't overcomplicate things. Just be strategic in how you assess and deduce what is taking place. All right, so let us try to do a blood group assessment. So let us say we have patient X, or let us call him patient 2, being that is a group 2 discrepancy. And then we have forward, FORWARD, and then we have reverse, REVERC. So we're going to use the anti-A and the anti-B. And then we're going to have the A1 cells, and we're going to have the B cells. So this patient has no reaction in the anti-A. It has a 1+ reaction in with the anti-B. For the A1, there's a 4+. For the B, there is a 4+ as well. The first thing that I want you to always remember is that if you get a 3 or 4+, that is usually the indicator as to the correct blood grouping system. All right. So when we see a 3 or 4+, we're knowing that this is highly likely that this is a true reaction. So especially in situations where we have a 1+ reaction or a 2+ reaction, that there is suspect or questionable. What we're going to try to do now is to figure out why is it that this group is saying O, but this group right here is telling us that this patient is B. There is a discrepancy, B and O, all right? But this is a 1+, so maybe this here is some reactivity that is taking place that should not be taking place. And if we wash these cells and then re-perform the blood grouping, then there's a high probability that we will have this resolve, and then patient 2 will have no reaction, and a 4+ reaction, and a 4+ reaction, which would give us group O. So something like that can give us an idea as to a discrepancy that occurs in the forward. All right. Sometimes we have type 3 discrepancies, and type 3 discrepancies are common. They're problematic. They are discrepancies that most persons don't like to have to deal with. All right. So, as we stated before, that type 3 discrepancies are between the forward and reverse grouping and are caused by protein or plasma abnormalities and result in rouleau formation. So, or pseudoagglutination, attributing to give us a reaction that looks like an agglutination, but we have to take that under the microscope to evaluate to see what is what. So, a rouleau formation gives us a situation of the blood cells stacking on each other, giving us a reaction that shouldn't be there. So we have to do a microscopic examination, or in situations like these with the type 3 discrepancies, what we can do is a warm wash to get off the excess protein that is causing the cells to stick together. All right. So let us do a quick example. Patient patient. We're going to call this patient 3 because this is a type 3 discrepancy, or forward as usual, forward grouping, and we're going to do a reverse grouping. All right, so we're going to use the anti-A, anti-B, the A1 cells, and the B cells. All right. So this patient had a 4+ reaction in the anti-A, 4+ reaction in the anti-B, then they have a 2+ reaction in the A cells, 2+ reaction in the B cells. This is problematic because this here is saying that the patient is AB, and this here is saying that the patient is O. But when we evaluate the strength of the reaction, this reaction is lower. So the reverse reaction has a lower strength. So we are going to try to resolve it from where the weakest point is, and that is the weakest point. So what we're going to do is we're going to examine for microscopically to see if this is a true agglutination. We're going to do saline replacement technique, and we're going to wash the cells with saline for three times at least, and then we're going to run antibody screen as needed to resolve this type of discrepancy. After we have done that, we realize that we got a reaction 4+, nothing, and nothing, which would give us the group of the patient to be AB. All right? Now, there is another kind of discrepancy which is considered to be the type 4 discrepancy. And we're just going to touch on the type 4 discrepancy a little bit. These discrepancies are between the forward and the reverse grouping. They are due to miscellaneous problem, usually maybe cold reacting auto antibody. in which red blood cells are so heavily coated with antibodies that they spontaneously agglutinate independently of the specific reagent being added to it to cause antigen antibody complex to form, right? So there is auto antibodies causing an agglutination. And when this happens, circulating red blood cells tend to give a lot of problems when we're doing the grouping, especially when we do it at room temperature. All right. So this is where the DTT comes in very critical for us to resolve this kind of discrepancy. All right. So don't be afraid of this type of discrepancy. This type of discrepancy does exist and it is solvable. All right. And if we observe a cold reacting, the first thing that I want you to think about after we have observed the reaction, we're going to try to wash the patient's cell with warm saline. And then we retest it, right? We're going to run DAT and we're going to run the auto control. We're going to run the auto control to see if there is auto antibodies. Then we're going to run the antibody screen as well. The DAT would indicate to us if there is sensitization to an external factor that went into the body, and the antibody screen would give us some indication of if the patient has any antibodies. All right. So that can be a possible solution. So let us try a patient 4, patient 4. And this patient, let's run that out, patient 4. And we're going to do the forward as always, forward. And reverse. So we're going to have anti-A, anti-B, A1 cells, and the B cells, all right? So what if we had a patient with 2+ reaction in the A, we had a 4+ reaction in the B, then we had a 4+ reaction in the A, and a 2+ reaction in the B. This here is confusing because this here, the forward is saying that A and B antibodies are present, which means that this person would be AB. And when we come to the reverse, A and B cells reacting, which means that this person is O. But the person can't be AB and O at the same time because you can't have the same antibodies to the antigen, because in that situation there is going to be a termination of the individual because agglutination would be taking place. All right. So next thing that we're going to look at, where is the weakest link or where is the weaker link? So this is a weak link because right here we realize that that's a 2+ reaction. Usually a 2+ reaction is indicating a problem if it's not corresponding. So these two areas, the A and the B, have a 2+. So if we take out the A and the B alone, does this give us the proper group? So anti-B and A1 cells. Yes, that would be a B group right here if this discrepancy is resolved. So what we're going to do is wash the cell with warm saline and we're going to do a retest. We're going to run the DAT, we're going to run the auto control, we're going to run the antibody screen, and then we're going to determine what the correct blood group is. After we have done that, guess what we came up with? We came up with no reaction, 4+ reaction. We came up with a 4+ reaction remain and no reaction. So this patient was actually group B. All right. So it's not that complicated as long as you know what it is that you're looking for. So let us try another one. This one should be pretty easy, pretty straightforward for you to be able to identify. So patient group 4. And we're going to do the forward grouping. And we're going to do the reverse grouping. So the forward, we have the anti-A, anti-B, then we have the A1 cells, and we also have the B cells. Now, we have a reaction in which we have 4 plus in the A, 4 plus in the B, we have 1 plus in the AB, and we have nothing in the B. This looks like the patient right here is AB. And right here, it looks like the patient is B. But we can't have A, B, and B in the same patient being expressed. And when we look at the weaker link, we realize that the A1 cells, where we have the reactivity, is a 1 plus. So what we could do is to say to ourselves, maybe this is a B person, but why is it that we're having some reaction with the A1? Is this normal? Yes, this is possible. We have seen this time and time again in our manual and automated methodologies, but this can be resolved. So what we need to do is assess the reaction with the A1 cells. Let us think of ourselves, or think to ourselves. You have A1, you have A2, you have A3, you have different subgroups of A. If we know that about 99% of the people are A1 and A2, and we have A1 being most prevalent, but there's a possibility that there is A2 that exists, then we're going to try to check with the A1 lectin to see if there is any reactivity with this patient cell. So we're going to test the cells with the A1 lectin. Then we're going to test the serum with the A1 and the A2. And also we're going to use the O cells, right? Then we're going to run the auto control. And then that should give us an idea that there is a possibility that this patient was actually A, B, but guess what? A2B, not A1B. So yes, it is that simple, but yet complicated simultaneously. So that is the nuances that exist. That's one of the nuances that exist in immunohematology. You have patients with different situation, different discrepancies. You have to try to figure out what is causing the particular reaction, so that there can give some persons a thrill if they are of an investigative mind. But before we depart, I just want to bring your attention to a situation where antibodies other than the anti-A and the anti-B may react to form the antigen antibody complex that may then be absorbed on to a patient's red blood cell. When we're thinking about type 4 discrepancy. So one example that I want you to just bear in mind is that of an individual with an antibody against the acryloflavin that is the yellow dye that is used in the anti-B reagent. So just bear in mind, in the anti-B, we use a yellow dye. In the anti-A, we use a blue dye. And that there is just for you to have a mental picture of the reagent. So the acrylflavin complex can attach to the patient's red blood cell, causing agglutination in the forward typing. So washing the patient's cell 3 times with saline and then retyping should resolve such a discrepancy. But in some cases, you have a cis AB, which refers to the inheritance of both the AB gene from one parent or one parent who is a carrier on one chromosome. And then guess what? You inherit the O gene from the other parent. All right. So this results into an offspring inheriting 3 ABO genes instead of 2. So they are inheriting the A, B gene from one parent, and then they're inheriting the O gene from another parent. So instead of having 2, they have 3. So this here is the cis AB. So this designation of cis AB is used to distinguish this mode of inheritance from the normal AB phenotype that we are accustomed to, in which the alleles are located on different chromosomes. The cis AB phenotype was first discovered in 1964 when a Polish family was described in which the father was group O and the mother was group AB and gave birth to children who were all AB. It was resolved by the fact that the A and the B genes were inherited together and were both on the same or the cis chromosome, thus the term cis AB. So yes, there are these situations that do occur, and the cis AB phenotype is a rare occurrence. An express weakly reactive A antigen and a weak B antigen simultaneously. The B antigen usually yields a weak reaction with the anti-B from random donors with mixed field agglutination, typically of subgroup B3, which has been reported in several cases. So the weak anti-B present in the serum of most cis-AB individuals lead to an ABO discrepancy in the reverse blood grouping. The serum of most cis-AB individuals contain a weak anti-B which reacts with every single ordinary B red blood cell. Yet, guess what? It does not react with the cis AB red blood cell. And with that said, it is fair to say class is dismissed. Have yourself a beautiful day.